No se puede trabajar cada uno solo en su feudo, ¿cierto? Sino que se debe hacer colaboración no solamente entre patólogos, sino que entre los médicos veterinarios que están en terreno, los patólogos que están en el laboratorio y las personas que están investigando a nivel básico las enfermedades. Por lo tanto, en ese espíritu, las personas que participan de esta fundación y las personas que participan de este grupo latino de patología comparativa y que tenemos distintos niveles de expertise, ¿cierto? Estamos dispuestos a ofrecer el distinto nivel de expertise, ¿cierto?, para poder expandir esta idea de conocimiento en patología. Entonces, cuando alguno de sus miembros, particularmente los miembros con más experiencia, como el doctor Lombardini, tiene alguna visita a los países donde estamos hablando, en este caso Chile, se ofrecen voluntariamente para dar alguna charla y extender algo de esa expertise y conectarse más con las personas en Latinoamérica, ¿cierto?, y en otras partes del mundo. Por lo tanto, el doctor Lombardini está acá para dar una serie de charlas para el Ejército de Chile en el aniversario de los cuerpos veterinarios de Chile, ¿cierto?, del Ejército. Y por lo tanto él encontró un espacio, nos escribió un correo y nos dijo si estábamos interesados en que él participara, a lo que por supuesto le dijimos que sí, por supuesto, por eso estamos acá, ¿okay? Entonces, el doctor Lombardini les va a hablar mucho mejor que yo, lo que yo puedo hacer acerca de sí mismo, pero él es médico veterinario, ¿cierto?, es diplomado en patología y él es miembro del Ejército de Estados Unidos, y gracias a eso no solamente ha participado en el desarrollo, ¿cierto?, de investigación de soluciones para enfermedades particularmente infecciosas, sino que también ha participado en el desarrollo de residencia, ¿cierto?, de la especialización en patología, y ha tenido la oportunidad de viajar de visitar distintos países, particularmente Tailandia, como salía en el preámbulo de la invitación, ¿cierto?, donde ha tenido la oportunidad de ver animales, ¿cierto?, de distinto tipo, y por lo tanto parte de su expertise y parte de su pasión, ¿cierto?, es la de los animales exóticos, y en particular hoy día nos va a hablar de la patología de animales marinos. Ahora, el doctor Lombardini tiene, no recuerdo si es un doctorado en España,
in Inghilterra,
pero estudió español.
no, ho studiato all'Università di Salamanca in Spagna e errore perché parlo italiano. Parlo italiano e hablo esperanto. Entiendo todo en español, ok?
Però ho
difficoltà di parlare spagnolo adesso. La mia mujer è italiana, per questo.
Por esa razón, dado que es mucho más fluido la presentación, ¿cierto?, en inglés, la presentación es efectivamente en inglés. Ahora, el doctor Lombardini obviamente va a hacer el esfuerzo de hablar un poco más pausadamente, ¿cierto? Sin embargo, si ustedes tienen alguna duda, están interesados en un tema en particular y no les quedó muy claro lo que dijo, o cualquier razón, ustedes levantan la mano y lo conversamos. Hay varias personas que podemos ayudar a traducir, ¿cierto? Si tienen preguntas, háganlas en español para que todos entendamos, ¿cierto? Y estoy seguro que esto va a fluir bastante bien, a pesar de que está efectivamente en inglés, ¿ya? Entonces, los dejo con el doctor Lombardini. Vamos a hacer la charla está dividida en una parte de tortugas marinas, ¿cierto? Después vamos a conversar algo de micropsia, y en el medio, más o menos a las 3:20, vamos a hacer un café para descansar. Si tienen dudas, podemos interrumpir en cualquier momento. Perfecto. Los dejo entonces con el doctor Lombardini.
Gracias.
It's very nice to be here. So I really appreciate the opportunity, and thank you, Professor Cifuentes. So, as Federico mentioned, I'm an officer in the United States Army, and I'm here on the invitation of the Chilean Army to celebrate the 120th anniversary of the Chilean Army's Veterinary Corps. Within my role as an officer and as a Veterinary Corps officer in the U.S. Army, I am very, very involved in medical research, specifically in vaccine and therapeutic development against tropical diseases. So my PhD is in malariology, specifically, which means that my day-to-day work is not related to exotic animals. However, it is my passion, and it's what I did prior to and throughout the course of my career to date, is working with exotic animals. A lot with reptiles and amphibians, an enormous amount with fish, and then also with marine mammals. When I was in the university studying veterinary medicine, I was part of an emergency marine mammal necropsy team, and we did all the work for a large part of the middle part of the United States, Atlantic side, for any strandings. We would go there and do the necropsies, collect the material, and then do the anatomic pathology and the histopathology associated with that. Then I studied in a program called Aquavet, which was with University of Pennsylvania and Cornell University, and 2 years of doing that, which was fish medicine and marine mammal medicine. I was stationed with the Army for 3 years in Bangkok in Thailand, and developed a very strong relationship, friendship, and collaboration with my Thai veterinary colleagues. And while I was there, I did quite a bit of marine mammal pathology as well. Now, that being said, all of you, with your extraordinarily long coastline, have probably far more experience with the species that you deal with here in Chile than I do. And I will welcome learning from you. All right? How many of you are marine biologists? Okay. How many are veterinary students? Okay, so the majority of you. How many pathologists? Excellent. All right. My job is to make sure that all of the veterinary students, by the time I'm done, decide to become pathologists. Right? I love pathology. And as Professor Cifuentes said, I'm a member of the Charles L. Davis Foundation, which has been operating for decades in international education of veterinary pathology. It's a non-profit organization. We are all volunteers, and we lecture. All of the lecturers will go anywhere when we're asked to lecture on a variety of topics in veterinary pathology. So I know that Chile has had other times when C.L. Davis has been here to lecture, probably on horse pathology or other examples, but it just was fortuitous. It was good luck that I happened to be here for the encounter with the Chilean Army and have this opportunity. Now, I'm very informal, regardless of the bow tie. So if you have questions, if you have difficulty understanding, I want you to stop me and ask questions. If I say something that's incorrect, I want you to stop me and correct me. Okay? But please, I really could learn a great deal about what your species are here. At the end of this, when I return to the United States, I have another week. We go then from here to Uruguay. After that, I come back to the US. I will send Professor Cifuentes a copy of the PDF of all my slides with links to good databases that you can use for marine mammal necropsy as well as turtle necropsy. So that way, if you don't understand the English, you can see the video, you can go back through my slides, you can see it again if you want to, if I don't bore you to sleep this time. And you'll have that as a resource, okay? Also, I will make sure that you have my email address, and I'm a resource for any of you. And if I don't know the answer, I will reach out to my colleagues who do, okay? All right, I have a lot of material, so we're just gonna push through it. And hopefully I won't put you to sleep, but we will take a break at some point in the middle. If you're dying, if you need to go to the bathroom, please get up and go. You will not offend me, okay? All right. Just so you understand, the way we're going to do this is I'm going to start off with a lecture on basic concepts of necropsy and comparative pathology. It's not going to be specific to marine mammals. You'll see a variety of different species, but I want you to realize you're all comparative pathologists. Any veterinarian who goes out there, you need to be comfortable necropsying anything. It doesn't matter if you've never seen it before. You can often apply what you know in your field of expertise to an exotic species, because often a lot of the anatomy translates. Sometimes there are very significant differences in anatomy and physiology, but the reality is, as long as you're open-minded, you can approach any necropsy with some level of confidence and be successful. Okay, so we're going to start off with just a brief lecture on techniques in describing the lesions. And then we're going to go into marine turtles. And there, I'm not going to spend time on necropsy techniques. That's going to be all classic diseases. And you'll see that all of my slides, there's very little words, if any. It's going to be mainly images. Okay? And I will ask you to be interactive. And then finally, we'll spend the rest of the lecture on marine mammals. Okay. All right. So, what's important to do is when you approach any species, you need to get a sense for what you're doing. You should know the anatomy. You should know what normal looks like. And that can be tricky. And I'll show you some pictures where I was caught off guard because I did not know what normal looked like and I thought I was looking at pathological lesions. You need to know the history, which is critical regardless of what you're doing in veterinary medicine. You should know the nutrition, know the environment. Try to gather as much data as you can prior to starting the necropsy. Okay? And then, this whole concept of being comparative pathologists. You need to apply what you know to what you're working with. So, I did my residency in veterinary pathology in the Army at a place called the Armed Forces Institute of Pathology, AFIP. That has since become the Joint Pathology Center, but it used to be sort of the ivory tower, or the highest level of expertise for pathology, both human and animal. That has changed for the most part, but it is still a very, very important place in terms of highest level of diagnostics. And what we did is during my second year of residency, we were assigned to the Smithsonian Zoo, to the National Institute of Health, NIH, and then to a diagnostic lab where we did large animal pathology. At the National Zoo, while I was doing my rotation there, I was required to do necropsies on Goliath beetles. I don't know anything about Goliath beetles. And this is complex. And obviously, I cannot apply what I know in a dog to a Goliath beetle in terms of anatomy or physiology or diseases. So there I had to do some quick research and started reading all the literature I could find to try to understand what I was doing. But eventually, I got pretty comfortable with this. On the other hand, here's a case. This is brain from a lynx, so it's a wild type of large cat. And what are you seeing here? What is the lesion? I think I heard it. There's ventricular hydrocephalus, okay? It's bilateral. But is this any different than what you would see in a dog or a domestic cat? No, it's exactly the same thing. I could have told you this was a domestic cat. It happens to be an exotic animal, but that doesn't matter. You know, sometimes the similar etiology can present in different species in a similar manner. So for example, pox viruses cause proliferative necrotizing lesions, regardless of if it's AV pox or seal pox, which is a parapox in this case, or a Lepori pox. They all cause more or less similar lesions when looked at under the microscope. In fact, you might have a hard time telling me the difference between these two, potentially. There are some differences, obviously, but at least in terms of gross presentation, they're similar. And so you can apply what you know from one species to another, at least in terms of trying to decrease your differential list. How about this? This is lung with some lymph node. I think it's mainly lung. So what's the disease, do you think? It's mycobacteriosis? Yes. Do you know what the species is? This is an Asian elephant. It's actually human TB. This is Mycobacterium tuberculosis, which is a big problem in Asian elephants, where humans have transmitted the disease to the elephants, and now they can give it back to us. How about that? This is also Mycobacterium. This is Mycobacterium bovis in a lion. What about this? This is a bird. And this is Mycobacterium avium intracellulare complex. All right? So, similar organisms, similar presentations in very different species. So this is a frog. And this is Mycobacterium marinum. Okay. So I was working with some students in Philadelphia, and we started doing a necropsy on a leatherback turtle. We took the head off, and it presented like this. I had never seen this before. But looking at this structure, which seems very, very alien. This is from some kind of horror movie. This is the esophagus. But I've never seen one of these before. I'd never seen it. Had not studied it, so I didn't know. But if I look at it, all of these look to be exactly the same, right? I don't see hemorrhage. I don't see any atypia in terms of that. So I'm going to assume it's a normal feature. And so yes, this is normal. This is normal esophagus. All right. How about this? This bullfrog. Where's the lesion? I'm going to start on this end. Oh, I cheated. All right. I'm going to start on this end, and I want you to stop me. Stop me when I get to the lesion. All right, so stop. So what is that? So it's fat atrophy. Those are the fat pads in the frog. So the frog was probably starving. And so it should look like this, which is basically just adipose and a lot of vascularity. But because of the starvation, this is what it looked like because of atrophy of the fat. So you have to know here what normal looks like to be able to interpret what the lesion is. Where's the lesion here? I'm going to start here. I want you to stop me. Did I hear IE? Yeah, that's where the lesion is. This is a congenital lesion in this clownfish. It's opercular hypoplasia. Okay, so the operculum was not fully covering the branchial arch and the gills, which is going to have some problems associated with it. The gills are going to become atrophied in that area that's exposed because of the friction and the as opposed to being protected. All right, it's a subtle lesion, but it's significant. How about here? So that's normal, and that's a fish that I caught. But this is a bass. So in the United States, in all the restaurants, we have Chilean sea bass. This is a freshwater bass. But this is what the fish looks like normally. Well, we got a nice congenital bulldog-like lesion. Okay. But it's a big fish. So it obviously survived pretty well, even with that kind of lesion. Okay. When you're doing a necropsy, it's important that you look absolutely everywhere. Okay, these are called gill maggots in a fish, on the fish gills. And if you didn't lift up the operculum, you can't see what's underneath here. So if you don't look under there, you're not going to see it. And this is an important point. Because with marine mammals, there's always a museum or a zoo or some organization that wants to preserve the whole skeleton. Have any of you done necropsies on marine mammals? Okay. Have you run into this problem where you're trying to do the work and they want to preserve the skeleton, so they won't let you examine everything? Yeah, it's a real problem for me as well. They ask you to do a cosmetic necropsy, which you can do, but that means you won't see the brain. You may not be able to see some of the other organs as well. This is a problem because you need to be able to do the best possible diagnosis you can of the entire animal. So you can still do a good job of cutting open the skull, removing the brain, and not overly damaging the body. But you may not be able to look at the spinal cord, for example. But this is a problem you will be faced with delicate diplomatic problem, but you also need to always try to push to be able to do a very complete necropsy when you can. So knowing your nutrition and the environment is very important too. This animal has albinism, so it's congenital, and I don't know if the deformation of the carapace is due to nutritional problems or if it's also congenital. It could be both. But nutritional problems in turtles will cause very, very significant changes to their carapace and to the plastron. Okay, plastron on the front, carapace on the back. So you need to know more about this. This is an alligator snapping turtle, which is a North American species. I don't know if you have them down in South America, but it's a very large turtle. It's a big animal. But this is wrong. This is not normal. Sometimes generalizing can help you, but there's certainly going to be very significant differences in anatomy and physiology which will impact the presentation of disease in a specific animal. This animal, an axolotl, lives in an aqueous environment, so it's constantly affected by its environment. Changes in that water quality can greatly impact it. In this case, nutritional changes, because this iguana probably was kept as a pet and did not have adequate UV light, or potentially dietary problems. And as a result, it's got a form of fibrous osteodystrophy, which we see in dogs, we see in horses. So if you saw this microscopically, or even grossly, you should be able to make some inference. Okay. With everything I've just said, being a comparative pathologist means that you need to be willing to generalize to a certain extent, at least to the level of the whole animal. And in this case, what is the lesion you're seeing? This is an American lobster, Pomaris, I think Pomar americanus, something like that. And it's got very, very severe changes to its carapace, right? Ulcerative changes. And the histology is going to be different here, but fundamentally what you're looking at is ulcerative necrotizing dermatitis, right? Fundamentally. Under the microscope, there's different tissue types, there's different organ structures, and you're going to have to use different terminology. But in general terms, if you're trying to describe this, you can probably use the terminology that you're comfortable with, which is to describe an ulcerative necrotizing dermatitis. Okay. One thing we used to say in the residency is you need to be able to describe, both microscopically and grossly, to a blind pathologist. Okay. And make them understand what you're seeing. All right. So I'm going to show you a couple of pictures in koi, both in koi, and the actual presentation, as you gain experience with certain species, you can start to see subtle differences that help you with the etiology, even though the same tissue is affected. So here the gills are necrotic, but I want you to look at the fact that it's you could probably draw a line around the affected tissue and healthier tissue. It's not all healthy, okay, but healthier. And that white area is significant. This happens to be Flavobacterium branchiophilium in a koi, which is bacterial gill disease. And then this one. What are you seeing that's different between this image and this image? They're both necrotizing. But what do we have here that we didn't really have in the other? Hemorrhage. Excellent. And this is koi herpes virus. So this is cypritid herpes virus type 3. This is a reportable disease, I think. Don't quote me on that. We'll just keep that in this room together, Mark. But it's an important disease that causes very, very significant virulence in koi, in carp, and a high level of mortality. All right, it's quite contagious. But the hemorrhage that you're seeing here helps you start to weed out the differences between the diseases and potentially start to think, I think this is viral rather than bacterial. In this case. It's also important to remember that the same etiology can present in different tissues. This is a type of bird from Hawaii. And this is avian pox, which you saw earlier with those lumps on that one small bird, but it can present in different places. This is also avian pox. Now, this bird may have trouble. Secondary bacterial infections, fungal infection, because this is going to start to ulcerate. It may not be able to perch or fly properly, but if the rest of the animal is okay, it can probably still feed. How is this bird? It's an eagle. How is this bird going to do? So you should expect to see some other changes associated with this, such as changes associated with starvation. Different etiologies occasionally will present with the exact same macroscopic presentation. So this is a beta, a Siamese fighting fish. So it's a freshwater fish, stagnant water really, from Southeast Asia. And these white spots, this is Ichthyotherius multifilus, or ich. And then this is a marine fish. And this is Cryptocaryon, irritans. Okay? They are similar species, but it's a marine versus freshwater. They are different species. They look similar. They present similarly grossly as well as microscopic. All right, this is a white-tailed deer from the east coast of the United States. So where's the lesion? I'll start here. I want a vet student to answer this. All right? Where's the lesion? Where's the lesion? Okay, do we have intestines in our thorax? What happened to this animal? It was hit by a car. Okay, so it's got a diaphragmatic hernia, and the intestines moved up into the thorax. This is not a particularly exotic animal, but it is still wildlife. But if I said this was a dog, it'd be the same thing. Okay? All right. How many eastern box turtles have you guys seen? So I found this turtle at my parents' house, which is in Maryland, on the east coast of the United States. And this turtle has a very severe case of ranavirus, okay, which attacks the blood vessels and causes very, very necrotizing disease in all of the viscera. But what are we seeing here? Where's the lesion? So, yes, and what is that? What are those white plaques, do you think? Well, I don't want an etiology, just generally, what are we dealing with? What's the process? Excellent, it's necrosis. So we have a necrotizing stomatitis, stomatitis for the mouth. It also happens to have damage to the nasal cavity and to the eyes. But you can describe this the same way, a necrotizing stomatitis, if it's a cat, with some of the lesions we see in cats' mouths, or in a species you've never seen before. Okay? Sometimes it's not easy though. Do you know what this is? This is the hemipene. So this is penile prolapse, or paraphymosis. Now we do get paraphymosis in a variety of domestic species, but this is a little bit unique based on the anatomy of the species. Okay. How many wombats have you worked with? You've worked with wombats?
Yeah, Australia.
Okay, excellent, excellent. But all right, all the rest of you do not. So she's the only one who knows what this is. No, you have this disease, we have this disease, we see it all the time. Scabies. This is Sarcoptes. Okay, it doesn't matter that
It's a wombat.
I'm going to show you a picture of a polar bear. And I've never worked with a polar bear. But I have a very good picture of scabies in a polar bear. So I found this also. So we have deer hunters who use bows and arrows. And I found this and then gave it to my professor who works at the University of Tennessee now. This arrow went in through here and lodged in the bone. But it didn't actually damage the spinal cord. And the animal lived with this in its back and was able to wall off through spondylosis, essentially, bridging of the bone between the vertebrae to stabilize. Okay? Now, this is a pretty unique lesion. I've never seen anything like this anywhere else. But conceptually, you see this in old dogs, right? In the military, we see it in old military working dogs all the time. Typically, the spondylosis is on the bottom here, ventral, but you can see dorsal spondylosis in dogs. You're going to describe it the same way. It's the same type of pathology. Even though you've never seen this kind of thing, and I'd be willing to bet that none of us will ever see it again. The pathology, the process through which the lesion developed, is going to be the same between species. Okay. So just some quick words of advice in terms of when you do your necropsies. I want you taking photographs. And I don't want you using your telephones. I want you to use a good camera. Because once you're done with the necropsy, what happens to the tissue? It goes in the trash, you burn it, and a week later when you're going through all your photographs and trying to think I've got a great publication here, I'm going to write this up, I'm going to publish this, your photographs aren't worth it. Okay? You're always going to regret that. So make sure that there's somebody there who can help you, or you yourself have a good camera with which you can take. Now iPhones are getting better and better and better, so eventually we'll be able to use just your telephones. But I always have a good camera with me, either my own or borrow one. If I know I'm doing, especially a marine mammal necropsy, you have species here in Chile. I've read the literature. I think the Chileans are the only ones who are publishing on blue whale pathology. And how many papers do you think there are? There's like 3. And why? Because we don't see them. They're whales that stay very deep, and they don't come close to the shore. And so if we're lucky, we get to see them. You know, I was talking to Professor Cifuentes on the way here, and he was saying that in one case there was a stranding that required the help of the Chilean Navy to get to the animals. And it took 2 months to get to them. But they were dead for 2 months. So you lose a lot of your diagnostic capability. So if you have that opportunity to do a necropsy on an exotic species like a blue whale, you need to make sure you're taking good photographs, because that is going to be, no matter what you find, it's going to be important for the literature, it's going to be important for all of us as the scientific community to learn from that. Okay? You need to take notes. They're going to get bloody, they're going to get disgusting, there's going to be formalin splashing on your paper, so transcribe them. But once you lose those notes, it's the same thing as throwing them in the trash. So unless you actually know what you've seen and you've taken good notes, you'll forget. But you need to be thorough and make sure that you're annotating and cataloging and describing everything. You need to read the literature and find out what's out there. I had a tumor, when I was a resident, I had a tumor in an axolotl. It was called a neuroepithelioma. And I was so excited about this. So excited. It was great. It was in the mouth of this axolotl. And I looked and looked. I didn't see anything in the literature. I finally found that there was a paper that had been written on exactly the same thing in, I think, 1920. I found it. But it was important to know that, because if not, I would have rushed forward and tried to publish on this. Or I may not have known what it was, but it's actually something that's been described relatively well in that species. But you need to know what's out there. You happen to know what this disease is? White nose disease. So Geomyces destructans, I think is the name. And this is wiping out the bat population in several parts of the United States, which is, as a person who studies malaria, it's a big deal. You need to publish. Who knows what this disease is? It's called devil facial tumor. It's a neuroendocrine, essentially, tumor of Tasmanian devils. You know how it's spread? By fighting. Do you know what other tumor that you probably have here in Chile would you compare it to? TBT. So this is one of those few tumors that are transmissible. Okay? But this is actually potentially, now I think they've been doing a good job in terms of conservation, but it came close to eradicating the species because they're extremely malignant tumors. But you need to publish this material. You need to publish. It's a pain. It is a pain in the rear end. It takes a very long time. You have to deal with a lot of punishing rejection, but you need to get that out there because we all need to learn from it. Okay. You have to expect that there's going to be emerging infectious diseases, even in Chile, where you are very protected, with the Andes on one side and the Pacific Ocean on the other side, and very small openings on both ends. You're very protected, which is wonderful. These last few days that I've been working and talking about tropical diseases like dengue and malaria and the rest, Chile is, you look at the map of the world of where these diseases exist, and it's all red except for Chile. And so the same is probably true for some of your animal diseases. I think you don't have foot and mouth disease, do you? Aphthovirus. Do you have aphtho? No? You eradicated it. But you're lucky because you have an extremely, extremely long and thin country. But you've got the Andes that protect you, natural barrier, and you've got the Pacific Ocean that protects you, natural barrier. Other countries aren't so lucky. Yeah, I know. But this is a real issue. So you need to consider the possibility of emerging diseases even in Chile. All right. And you have to get excited about pathology. It is one of the disciplines that provides you with the greatest possibility of exposure to different species. Here I am, I work with primarily laboratory animals, but I get to work with fish. Right now I do a lot of pathology for certain zoos, and so I'm seeing animals that I've never seen before, different types of snakes, birds, beautiful birds that, well, when they were alive they were beautiful, but I see the slides, and it's exciting for me because I get to see very, very unique diseases. Okay. Yes. Yeah, exactly. So it's a lice is a bit of a misnomer, but they're a sea parasite that will attach to different parts of the fish, often to the gills. They can present in different ways, and they look very, very different. A wide range of them. These are the reproductive organs, but this poor fish is not happy. Okay, we're going to jump right into turtles. Keep going. You stop me if you want me to, okay? I could talk all day. All right. These are some resources. You're going to get the PDF of this eventually once I get back to the United States, so you can check these out. I went and looked. These are wonderful sites. They're free. You can access them on the internet, and they have a beautiful, detailed description through organ by organ, system by system of how to do a necropsy in sea turtles. So don't worry about writing it down. You can if you want to, take photographs if you want to, but you're going to get this. I promise you. Okay? I see people writing. All right, can I move? You take a picture? There you go. And bam. Okay, we're done. All right. Okay. What is this disease? Fibropapilloma, excellent. Do you know what is the cause? Herpes virus, excellent. All right. So, in this case, this is an ocular fibropapilloma. These are associated with, at least it's posited to be associated with turtle herpes virus, which is causing an epidemic in sea turtles. Green sea turtles seem to be particularly affected by this. There is a recent report of a sea turtle tornovirus 1, which was isolated from an infected animal. Histopathologically, the characteristics that distinguish these tumors from other forms of neoplasia are both the combination of epithelial and dermal changes. So you'll see both a mesenchymal and epithelial element to this microscopically. And they've been reported in all sea turtle species except for leatherbacks. But as I said, green sea turtles seem to be particularly affected. Now, these can regress on their own. There have been a few reports of it metastasizing into the internal viscera, but a turtle could survive this depending on where the lesions are. So, this turtle is probably doing relatively well right now, assuming we don't have tumors all over the rest of the body. But this one is going to have some problems. Not because the tumor itself is causing direct damage to the turtle, although that's probably happening as well. But what do we have here? How do turtles feed themselves? Okay. They need to be hydrodynamic. And he's lost his streamlined nature in terms of being able to get prey, potentially, depending on the species. But this could be a big problem in terms of feeding. And we do see them a lot around the head as well. So that could cause blindness, it could cause problems in the oral cavity or around the oral cavity, which can impede that. And then we also have to worry about secondary infections associated with any kind of large tumor like this. Okay. So you see all of the tumors that we see here? And there are millions of photographs associated with this that are available if you want to search the internet for this particular disease. It's a very common disease, unfortunately, and is causing a great deal of a problem with, especially the green sea turtle. You can see a lot of this in the Hawaiian waters. All right. So remember the lobster. What are we looking at here? This is a necrotizing and ulcerative dermatitis. It's probably extended even deeper there. These are some pretty advanced lesions. And certainly I would expect to see secondary bacterial or fungal infections in an animal like this. Both land and sea turtles will develop diseases called shell disease. Now what the actual etiologies are that are associated with a particular disease in a particular species may change. In sea turtles, we see Benechia chitnivora as a bacterial agent which will affect that. It's a common infectious agent of crustaceans, but it's seen as causing erythema and pitting of the carapace in sea turtles. In land turtles, we'll see Citrobacter frundii and seriata species causing similar diseases. We'll see it in freshwater turtles as well as in land turtles. And I have some pictures that I'm not going to show you here, but of soft shell turtles from Thailand with nice lesions that was associated with Citrobacter. So in these animals, you should expect to see anorexia and lethargy, and then petechial hemorrhages, which are essentially cutaneous infarcts in the shell and the skin. And then there'll often be liver necrosis also in these animals. Okay. So with chronicity, some of them can heal, but often they'll present like this. This is caused by bacillomyces, and this is a hepatitis, so this is the liver. This is a hepatitis. It's necrotizing in granulomatous. And there actually happens to be a vasculitis in this. Now you wouldn't be able to see that grossly, although you can make some interpretations based on presentation. That's a subtle area that typically you don't need to worry about. It's going to be more for the pathologist to worry about. But for example, if I see nematodes, parasites, in the intestine of an animal, any animal, and I'm describing that, I might suggest as part of my description that there's an eosinophilia associated with it. I know that they're there. I can't see them. That's the delicate area of the balance that you have to make. I know they're there because of the fact that the lesion is associated with the parasites. Okay? And so in this case, because of the distribution, we can think, okay, well, how is this happening? How is this spreading? And if it's spreading through the vasculature, then you might consider this to be embolic, or you might suggest that it has a vasculitis associated with it. Yes?
What was the agent of it?
It's Bacillomyces. Okay. And of import here is that this is a fungal disease. So what do we know about fungal diseases? What does the zygomyces do in a cow's rumen? You know how it spreads? So you know how you get a ruminitis? Maybe because of illness and you get a change in the bacterial content, the pH, and then as a result, maybe the acidity rises, you get damage to the mucosa, and then it allows for fungus to actually invade in. How does fungus travel in that case? Because you'll see it then in the liver and places like that. How does it get to the liver? It gets to the liver because the fungus likes to invade through the walls of the vasculature. All right, and then it'll spread just the way you get through a septic process, through the bloodstream. Okay, typically I have separating slides between the different etiologies. A lot of pathologists will lecture by system, but as you can see, I lecture by etiology, by group of etiology. So we're now in parasites, okay? And this is a really interesting disease that I've seen quite a bit. I hope to publish this, but I was involved in a series of necropsies of exotic turtles and tortoises that were smuggled from Pakistan and India into Bangkok. And the intent was that they were going to go from Bangkok probably to Europe or the United States or China for the pet trade. This is a big problem. And Bangkok, unfortunately, is sort of a clearinghouse for animal smuggling. But these turtles, a lot of them died. There was a man who came in with suitcases, and he had about 300 per suitcase. He had several. In the end, I think there were almost 1,000 animals. And these turtles were taped and just packed in like sardines. And a lot of them died. And so we did necropsies on the ones that did. And this presented a few problems. First of all, we saw all sorts of parasites. Okay? These were pretty unhealthy animals anyway. They were immunosuppressed and stressed associated with their treatment. But what was interesting is I was seeing parasites that should be localized to the Indian subcontinent, Pakistan, India area. But instead, we now have them in Thailand, and they were intending on going all over. Now, these weren't zoonoses for the most part, so it may not have impacted human health. But you can think of what happens if this gets into a wildlife population, where the animals are naive. It could wipe out an endangered species easily. Okay? And so this was something, this parasite is something that I saw in some of those turtles and tortoises. This is called Liridrius liridae. And I'm probably saying that wrong. I'm not a parasitologist, so I'm going to butcher a lot of the parasite names. I apologize. And these are cardiac trematodes. Okay. In one case, there was a black turtle from Mexico that was reported to have an invasion of these spiroorchid trematode eggs, because it's not the adult necessarily that's causing the primary damage, but when they lay eggs, those eggs end up traveling through the bloodstream and cause granulomas in a variety of tissues. So in one of these tortoises that I necropsied and did the microscopic work on in Thailand, we had lesions throughout the body. Almost every tissue was affected. But we'll see this in a variety of species. So in that particular animal from Mexico, we had the trematode eggs in the liver, the kidneys, the intestines, the muscle, the heart, pancreas, and the duodenum. There were 75 adult trematodes were recovered from the heart of a single animal. Well, we see a lot of this also in the marine turtles. There's going to be a range of different species of trematode which will act in similar ways. So, you know, the Mexican land animal probably didn't have the same species as we see here. And certainly what I saw in those tortoises from India was not the same, but the pathogenesis is similar. Okay, and these are trematodes within the stomach of a green turtle. These are the adults. This is something called marine leeches, and there's two types of leech that feed exclusively on the blood of turtles. Both are called Ozobranchus species, and they're found along the Atlantic coast of the United States and into the Gulf of Mexico. And what's interesting about this is the Ozobranchus branchiatus specifically targets green turtles, while Ozobranchus mardui is seen in a range of turtles, but they typically go after the loggerheads. However, the first one is only the green turtle specific. They typically attach themselves to the underside of the flippers, to the mouth, to the neck, to the cloaca, and they can cause both ulceration associated with their physical presence there and the way they're feeding, but they can, and so secondary infections, but they can also cause anemia as well, depending on the amount that you have. There have been reports of epizootics of these leeches that can result in even the death of infected animals. This is a process, and we're going to talk in general terms about this. This is called epibiota, and you're going to see this also in the marine mammal talk. We see this in whales, certainly, but in a range of other cetaceans. This is a unique characteristic. We see it often in loggerhead turtles, but you have an entire community of organisms, or an epibiota, that live on the shell of a single animal, almost a little microcosm that exists on that one animal, a different environment. In some cases, we've seen over 80 different species of animal, of invertebrate that are living on a single turtle. Okay. Now, sometimes it's just going to be barnacles, but it should raise suspicion. The turtles aren't necessarily accumulating this because there's a benefit to them. And typically turtles will do a pretty good job of keeping themselves free of excessive growth, although in loggerheads you can see certainly the barnacles growing. So you should be suspicious of immunosuppression or some other problem occurring in an animal like this. Okay, tissue, what was it? Tissue from a red-tailed boa. So this is not a penile prolapse, but this is a rectal prolapse in this animal, and I don't know the cause in this particular case. Although, how do turtles reproduce? Are they live bearing? Are they live bearing? Do they bear live birth? No. Somebody said no, but they're egg. They lay eggs for the most part. Can't think of any turtles that are live bearers. So they lay eggs. So certainly you can get turtles that are egg bound, and that could cause stress and straining, which could result in a lesion like this. But I don't know the cause of why this animal prolapsed. Okay. There's nothing unique about this, but you should expect to see these kinds of lesions. So if a turtle lays thousands of eggs, then over their lifetime, they may only expect to see 1 or 2 offspring survive to adulthood. I worked I did a bushmeat study before, right after vet school, in Equatorial Guinea, in West Africa. And I spent some time on the volcanic beaches where a lot of the turtles lay their eggs. And we were doing counts, nest counts, at midnight on volcanic beaches under the moon. But while I was there, we were seeing, I was learning a great deal about what was also happening. And there was a specific group that had come into the country that, as part of their culture, wanted turtle eggs for a special celebration. And unfortunately, the army of that country went in and was harvesting the eggs directly out from under the females as they were laying them. Or they were actually taking the females and killing them for use for food. But what they're doing is potentially completely damaging the species, because especially if you're taking a turtle off the nest before it's had a chance to lay, and you're killing it for food, well, you're wiping out the possibility of the next generation. But predation is a big problem with turtles. It's not necessarily pathology per se, but it's something that you need to expect. Okay, cold stunning is something which we saw quite a bit of this year in the United States because of atypical weather patterns that have started to affect us in the north. And so cold stunning refers to a hypothermic reaction, and it typically happens when the turtles have been exposed to a prolonged period of very cold water. The initial symptoms can include a decreased heart rate and decreased circulation and lethargy, and then they can go into shock, they can develop pneumonia, and they can often possibly die. Okay, and this is believed to be associated with climate change. I don't think I need to explain this. Okay, this is a very unfortunate problem, and certainly you have an enormous fishing industry, as do we. And so, depending on the type of fishing, if it's fishing that is sort of wholesale without distinguishing between species, it can catch up a lot of turtles as well. And we talked about poaching. Okay. All right. Do you want to take a break? This is the beginning, so from here on it's marine mammals. Okay, how am I doing? I've done 1 hour and 10 minutes. We have, who votes for a break? It's a democracy here. Okay, I think we have a majority. All right, let's take, how much? How much time? Yeah, any questions?
Yo he visto una foto de muchos reptiles, he visto cerosas negras.
So you saw a photograph of a necropsy of a reptile?
Yes.
With something black? I didn't understand. Here, I can yell.
He visto fotos de necropsias en otras especies de reptiles y he visto que algunos tienen la cerosa negra.
Black cirrhosis?
Yes.
Of the liver? I've never heard that term, black cirrhosis.
No cirrhosis.
Es la...
That everything turns black? Let me do some research. I'm not aware of that, but let me find out and I'll get back to you. So, the gentleman taking the photograph over there.
Sure.
There he is. If you have a question for me and I need to get back to you, either through Professor Cifuentes or you can write it down, give me your email, I will respond to you, okay?
¿Alguna pregunta?
Am I speaking too quickly?
No.
You understand me?
Yes.
All right. As you want. For the rest of them.
Hola. Yo quiero preguntarte los cambios patológicos más aparentes en tortugas que sufrieron hipotermia, que murieron, ahora que está diciendo, porque aquí recibimos mucho tortugas final terminaron parando en las costas. Pero es...
Parando es stranding?
Yes, stranding.
Okay.
Y muchas llegan ya prácticamente letárgicas y muertas. Quería ver qué experiencia tienen ustedes en Estados Unidos, porque allá es diferente, allá es el cambio de corriente fría que hace que las tortugas se queden atrapadas en ese sitio frío y entonces...
So we were seeing a lot of flash periods where it just changed very dramatically.
And so you all heard the question? She was asking about cold stunning. So I've personally not worked on necropsies of cold stunned turtles. It's a recent problem. It's a new thing that we're seeing happening more and more. As I mentioned, typically you're going to see some clinical changes first. So anorexia, lethargy, decreased heart rate. But then what you can see is because of those clinical changes, you can see aspiration pneumonia, which would probably be a big part of it, maybe some malnutrition, and then shock, typically, and death. So I don't know if there are specific pathological findings that have been described thus far. I've not read of any, but I think when we get enough data together, that might make for a very good paper, especially if you have the same thing in Chile. Any other questions? Okay, how long of a break? 20-minute break. Thank you. This is going to be the bulk of the talk. There's another 100+ slides, okay? But it's going to go faster once we get towards the parasites and the miscellaneous diseases, because I'm not as worried about you knowing specific parasite names. You can always look those up. But let's dive in. And Professor Cifuentes let me know that a lot of you are very interested in the whole concept of the actual logistics of the necropsy. Is that correct? For the most part? Okay, so we'll spend a little bit of time on that, although I'm going to make a few fundamental points, which should give you some guidance. So by the way, this is an Indo-Pacific humpback dolphin, the pink humpback dolphins from the Gulf of Thailand. They're beautiful animals, and I'm going to unfortunately show you some very severe pathology. Okay, so there is only one way to do a necropsy. That's a complete lie. There isn't. Okay, and I don't want anybody to ever tell you that. Now, the marine mammal community has very strict guidelines in terms of the data they want you to collect. But you're going to do the necropsy however you think is best, as long as you're thorough and you touch on everything. I don't care how you do it, as long as you're thorough. Now, what I'll do is I'll include with the material that I send Professor Cifuentes, I'll send some examples of some of the data collection sheets that are used by places such as Sausalito in California, and some of the other stranding networks in the United States, where they collect specific data. As a pathologist, I'm not as worried about measuring blubber thickness in all the different areas. It's not as interesting to me. It's important, though, because it helps with population dynamics, population biology, and it gives you data in terms of all of the species that you see. It's even more important when you're dealing with animals that are more exotic, that are rare, that we don't see as often. But the reality is, how you approach a necropsy is primarily up to you, as long as you are thorough. And part of that being thorough is fighting with the museum that we talked about, so that they let you look at the brain. I can't tell you how many times I've had people come in and try to take the skeleton away from me as I'm trying to do a good job at diagnosing disease. And I know it's important. The skull morphology and all of that is critically important, just as the blubber thicknesses are, in terms of giving the scientific community a true understanding of populations. But the reality is, the pathology is just as important, and we often see lesions in the brain. Okay? So, the way you can approach a necropsy, the way I personally approach a necropsy is obviously going to depend on the species. So one thing I might do is, after I've taken measurements, I like to go in through a ventral approach. But if it's a big animal, you can't do that. If it's a whale, you're going to go in wherever it's stranded. I don't care if that's standing on its head, you're going to try to figure out a way of going into it. But if you have a choice, if the animal is brought into you into a rehabilitation center and you're able to do the necropsy on a table and in a more controlled environment, then either it's going to be a table that will allow for the dorsal fin to fit into something, and so you can look at it ventral dorsal, or you can look at it on the side. And you can go in and create a window through which you can approach the organs. Now, one thing that is going to be important, I'm going to show you some pathology associated with it, is, so here, the abdomen and thorax are going to be, you see it here, okay? Starts there. Does that mean you don't look at any of this? You've got to cut through the blubber here. You should bread loaf the blubber, because you're going to see parasites often. Okay, and it's important to look at those. But all the vet students, you have done necropsies on something, on a dog, right? There's going to be some anatomical differences, but for the most part, the organs are the same. Okay, it's going to have a liver, it's going to have a spleen. The kidneys are going to look different. Okay, the lungs are going to have more cartilage. So they're going to have more cartilage because these are diving animals that have pressure differences that they have to deal with. But realistically, you shouldn't be surprised by pretty much anything you see in terms of the normal anatomy. That's not a surprise. All right, typically the way I do any necropsy is I'll open the abdomen first, carefully, and then I'll sample. So I'm going to look in as sterilely as possible, and I'm going to sample if there's any fluid in there, so that I can culture that if there's an effusion. That's why I like to do my necropsies ventral dorsal, because it keeps everything inside, as opposed to if you open it on the side, then you might slosh everything out. But it's a nice way to start. And I like to look at everything in situ. So I'll open up the abdomen first and create a window. I'll get a good idea of what I'm looking at. Is anything missing? So I want to look at everything in situ. Just like that deer. Do I have intestines in the chest? Shouldn't be there. But I will look, so I'll look at both kidneys. I'm going to show you some congenital lesions later on that'll show differences between the kidneys. I'm going to look to make sure I have everything, first of all, and then everything appears to be in the right place. Stranding can be a very violent process, especially if there's rocks. These animals get thrown about. If they die in the water, then they may, or even if they're very ill in the water, they may have gone through a lot of trauma by the time they hit the shore. So I showed you the shark lesions on the turtle. You should expect to see predation on the cetacea as well through the process. But remember, if they've gone through a traumatic process of getting to shore, they may simply have abrasions that are associated with hitting rocks or even the sand. And you'll know that once you pick up a dolphin, you can actually create lesions in it just with your hands once they start to dry. Have you experienced that? A little bit? Okay. So, but you should look externally. Make sure you're cataloging everything that you see, photograph everything. The marine mammal community is going to want you to measure the blubber at different sites across around the body. And then when you're ready to go inside, I want you looking at everything in situ, and then have a method. It's your method. But have a structured method through which, even if you're not thinking, you're going to hit every organ and collect every organ that you need, because you need to look at everything. So I told you my PhD is in malaria, and I know this is completely off topic, but I did a comparative study between a specific type of monkey malaria and human falciparum malaria. And I looked at over 100 years worth of human data, human autopsy data, from people who died from severe malaria. And there is no data on the spinal cord. Why? Were they lazy? I don't know. But I collected the spinal cord in every single animal that I did the necropsy on. Which is another problem associated with the museums, because they do want the entire skeleton. So maybe you just want to take samples. You might be able to separate the vertebrae and just cut out a sample from the inside so you don't damage the bone. You've got to compromise to a certain extent, but you need to be looking at everything. So collect everything, have a method, and make sure you get everything into formalin, everything photographed, everything annotated, because then you will have done a good job with it. Most of my experience is with cetacea that are manageable. Dolphins, porpoises, seals, sea lions, pygmy whales, that are not, and then a couple of whales that are maybe, this big. But nothing, I've never done a humpback whale or a sperm whale or a blue whale. Those are a whole different experience. And those require teams of people. So from doing elephants, which I have done, you also have teams of people, because you simply can't try to manage it by yourself. And so typically what you do is there's going to be a person in charge. And that person is going to dedicate teams to different organ systems to be complete. Now, one thing I want to warn you, if you're working on a smaller animal, avoid having more than one knife going at the same time. There aren't too many diseases that we can transmit back and forth between marine mammals and humans, although there are some. But I'll tell you, when I was in charge of doing monkey necropsies, there was only one knife working at a time because of the danger of us stabbing each other. And you just don't want to risk that. So just be careful about that. Have situational awareness. The person's in charge. Maintain situational awareness of what's happening. If you're working on a large cetacean, then dedicate one crew to looking at the reproductive tract, one crew to look at the digestive tract, one or maybe multiple crews to look at the digestive tract, one crew to look at the respiratory tract, etc. Okay? All the way through. And then have a whole separate team that's trying to get into the head. And it's the same thing with elephants. All right. But otherwise, you're not seeing too many anatomical differences. The blowhole is an anatomic difference, but it's really an extension of the upper respiratory tract. You need to look at it though, okay? Because there are diseases that will enter through there, and so you need to make sure you're looking at that. And you have to look at the brain. Okay. I'm sorry that I'm not giving you more detail. I will provide you with some examples, but the reality is there is no one way to do a necropsy. There is no right way to do a necropsy as long as you are thorough. As long as you collect everything. Okay. Any questions before I move on for that? Because I know that's an important subject for you. Yes?
Yeah, in regards to, we were talking about knives, right?
Yes.
So I'm curious on what other tools you use for these.
Okay.
You mentioned your hands, right?
Right. So you need to get into the brain. So if it's a dolphin, I'll use a hand saw. You don't need special tools really. I'll use a hand saw, maybe some screwdrivers and a small hammer. But I'm going to saw. I'm going to do a cut like that and like that on both sides, all the way across. Pop out that sort of diamond-shaped piece, and then I'll work in and around to get the brain out. I want to see the brain intact, if I can. What am I going to look at once I've got the brain out? What's still inside the calvarium? The pituitary gland. Because you're probably not going to have that come out because there's tissue that covers it. It sits in the tursa celica. Tursa celica. I forget. But you need to cut that out. You need to look at it. You can have lesions there, potentially. You want to look at the optic chiasm. Okay? The nerves that are going to cross, because those often stay in the calvarium as well. But you need to take that out, look inside, and then you can take that nice piece that you've cut and put that back. Okay, so to answer your question, sorry. So a handsaw, you need enough formalin. So you're going to have multiple jars. Typically you're going to have maybe some tags that you can attach to certain sections of, say, the intestine, or maybe little cassettes that you could put smaller organs or slices of smaller organs. A cutting board is a good thing. Multiple cutting boards, because what you can do is if you have a team, then you collect as the pathologist or as the main person in charge, you collect the material and you can hand off sections. Here's the liver, and you have somebody in the back bread loafing the liver and then collecting representative samples of that and photographing that. It's more efficient. A necropsy can take a very long time, or you could do it pretty quickly, depending on how efficient you are and how regimented you are in terms of the method that you use. Scissors are good. As I go into the brain in a smaller animal, I'm going to have to cut a little bit of the connective tissue between the meninges and the brain itself. I might have to cut some cranial nerves, because I'm going to work my way around, and then I'm going to use gravity to help me too. And so you might have somebody helping you as you're working your way in to cut the tissue out to get the brain out as a whole piece. Dolphin brains are big. You know, they'd be like this. They're not small.
They have bigger brain to body mass than we do. So you may need help with that. Then, if you're going to have to get into the thorax, and depending on if you've got somebody from the museum looking over your shoulder, then you can actually use some garden shears. You know what I'm talking about? To cut wood, to clean your garden. Look, you can go to the hardware store and buy everything you need. It doesn't have to be anything special. Yes, you can get a striker saw, and you can do things that way. But if you have a field necropsy kit, and I keep a field necropsy kit in my garage, it's ready to go at any time, and it's got some basic tools in there, and they are hardware type tools, and I can get everything I need with some basic tools in a smaller one. Now, a larger animal, a blue whale, you may actually need a chainsaw. Okay, you may need bigger instruments to be able to get in through the bone. But for a dolphin, you can use some pretty minimal. I will tell you though, one thing that absolutely is important is a good camera. Okay? I can't tell you how important that is. And it's in large part because once you're done, you're done. You can't go back and get more. Okay? Because it's burned, it's been given to the museum, it's in the trash. You're not going to go dumpster diving. You know what that means? That's a term we use in the United States. A dumpster is where you put all the garbage. It's a big metal container. You don't want to go diving into a dumpster to go get pieces of whale afterwards. So try to do everything efficiently the first time. You have to take good notes. So I typically carry a pencil with me because water and formalin will wash ink away. I'm not going to bring something valuable, so you can do a lot of this first right there, and then you can transcribe it later. You could have somebody taking notes for you that's clean. You should have gloves. You should have proper PPE. So maybe 2 sets of gloves per hand. You should have some mouth protection, some eye protection. Okay? I recommend, if it's a larger animal, that you're wearing heavy-duty boots and clothing that you don't care gets really, really bloody. Doing an elephant's the same thing, although you've got to be very careful because of tuberculosis. But you've got to get in there. In a large whale, you're inside the whale. And it's going to be disgusting. But that's the beauty of being a pathologist. Okay. So a lot of it's going to be based on your own technique. I personally work with very few tools. One thing that is going to be important, because the blubber is thick, is either you have several knives or you have a stone, okay, to be able to sharpen while you're going, because you're going to dull that knife very quickly. But really, there isn't too much else. You will need enough formalin, so you need jars and formalin to be able to put this stuff in. I take a lot of tissue. Because I can't go back for more. So I take a lot of tissue. Okay? And then you need trash bags and stuff like that. Yes? Yeah, absolutely. If I have any thought that there's going to be virus, then I'm going to take some samples in little bags, in quick bags for maybe PCR work. I can deep freeze those. Do alcohol as well. You know, one big problem that I see a lot of, is that I like to do electron microscopy. But you need special fixation for electron microscopy. So I typically will prepare and have some glutaraldehyde with me. You know, that might be a bridge too far, might be too much. But if you can, if it's being done here at the university, then you might consider collecting in all of those different modalities. And from a viral standpoint, yeah, it's important to be able to do PCR, do molecular work. Parasites, you can collect in alcohol so that your parasitologist colleagues can then look at the adult worms. There's some microscopic parasitology is a whole different story. I could give a whole 8 hours just on that. But collecting parasites in alcohol is a good idea if you see them. Representative samples. You know, what else do you use? Sorry to put you on the spot, but you're another pathologist. So what else have I missed? Anything I've missed that you can think of? Maybe some rat tooth tweezers to help you, something like that. I use 10% buffered formalin. I'm 253 years old because I drink formalin. No, you know, I've always used formalin. It is dangerous. You have to be careful about that. And whoever's in charge needs to make sure that the formalin is being kept away, and you're not in a closed room, hopefully, and that the samples are being taken and put over there. But that's again why you might have multiple cutting boards and people working on those. So I did a study. I haven't published it yet, but I'm going to. This is again just between us, okay? For the camera. I've done a lot of work in very austere environments, in parts of the world where I don't have access to much, and so I've collected controlled tissues from an animal and fixed them in a wide range of different possible fixatives that are commercially available. And what I'm in the middle of doing right now is reading out all of the different tissues and cataloging all of the artifact that's associated with those. And it's specifically for the use of a pathologist who might be in a very, very difficult area. You know, if you're in the middle of the Andes looking at necropsies on some species where you can't easily get back to Santiago or somewhere else to get more formalin, and you run out of formalin, you might have to look for something that you can buy locally. You know, and that could be isopropyl alcohol or something else. But I'm looking at all of those different things to try to compare. You know, it's not going to be this is not a study with molecular technology or the rest. It's just going to be an anatomic pathology assessment of artifact based on different fixatives in a controlled manner, because it was the same animal, you know, each different, the liver, one here, one here, one here, one here, and all the different fixatives. So we'll see. We'll see how it comes out. But it is a problem. Frankly, formalin is best. But for electron microscopy, you need glutaraldehyde. You can use it from formalin samples, but they don't do well. So, does that work? Did I answer your question, sir? We're good?
Yeah, I was thinking about the hooks or something.
Butcher hooks or something like that?
Hooks?
Yeah, because
I assume the skin's slippery and the blubber and everything.
If they dry out quickly. Yes, when you cut through the blubber, you're going to have fat on your fingers. But I bring a lot of gloves, and I change gloves a lot. You do? Yeah, for big whales, yeah, I'm sure. Well, also because it'd be so heavy to be able to do that. But I have not done big whales, so I don't have that direct experience. Okay, any other questions in terms of this? Yes?
Hi. I just want to know, what do you think about using prophylactic medicines? Because you work all your life doing this.
You mean for me?
Yes.
I don't know what medicines there are that I could take prophylactically for. Are you talking about the fixative, the formalin, or
no.
against diseases?
Preventive, for example.
Okay, I understand. Against zoonotic disease. Well, in marine mammals, there's some bacterial diseases that would be of concern. There's a fungal disease that would be zoonotic. There's really not much in the way of viral diseases that I worry about. Seal finger, so seal pox, yeah. Well, okay, so for seal pox, I apologize, you're right. That is zoonotic. But any other viruses? I can't think of any. Well, also seal pox can be zoonotic. It has been shown to happen in people who work in rehabilitation centers. So that's a pox virus. But mycobacteria, yeah, you're going to worry about. I've never taken anything. Personal protective gear should take care of all of that. So if you're careful and you protect your eyes, your mouth, you wear a mask. It doesn't have to be a PAPR. It doesn't have to be something with oxygen being pumped in. And and boots and gloves, and you take a good shower afterwards, I think you'd be okay. I don't want to be responsible for your health, but I haven't gotten sick.
No, I'm asking because we have some recommendations about, I don't know, maybe anti-parasites or...
Yeah, again,
like that.
How are you going to contract the parasite? So if it's a hookworm, you could get it just from walking on it, but that wouldn't be from a dolphin. If it's a cesto, you could get, there are some cestos, like anisakis, that you could get from a marine mammal, but you'd have to consume it.
Yes.
And really, that shouldn't be happening. So, I personally, it wouldn't be something that I would personally do. I can't speak to the regulations, though, otherwise. Yes, Professor.
Del punto de vista de alguien que trabaja en el laboratorio y recibimos el frasco con los trozos de tejido, the necropsy in a jar.
Yes.
Entonces, ¿qué importancia le das al tamaño del trozo? Porque esta muestra la toma alguien en la playa. The sample is taken by someone on the beach. El animal ya lleva muerto un par de días y después llega un pedazo de tejido de este porte, a big piece of tissue inside of a...
Okay, so I'm glad you mentioned that because that is important. So you have to allow for formalin to get into the tissue. You could not take the entire brain and dump it into a bucket and expect that it's going to be good fixation. Already, autolysis is a major problem for the histopathologist in reading under the microscope tissues from marine mammals, because autolysis is a fact of life. But you certainly are making things worse if you give big blocks of tissue and put it in, a couple drops of formalin on top. And that's what we see a lot. Necropsy in a jar happens everywhere in the world. See it all the time in the United States too. The clinician takes an entire dog and into a jar, a test tube. Obviously, that's not going to help because the formalin's not getting in. So, when you have your technical staff or the other veterinarians or whoever it is who is helping you, as they're bread loafing, they're taking small samples. It doesn't have to be just a small 2-centimeter cube. It can be bigger pieces, but they should be thin if they're bread loafed. Make sure they're getting representative samples, and that those samples, if it's pathology and you can see it grossly, you try to put some normal tissue next to it. So if you see a tumor, take the edge between the tumor and some normal tissue so they can identify where you are. Then after 24 hours, you should change that formalin because the blood, I believe, inactivates the formalin. So before sending it to the laboratory, ideally you've done all of that. You do need to, he's absolutely right. You want to be careful about that piece of not putting tissue pieces that are too big. I don't know, you can look up on the internet the specific distance, the specific amount that typically formalin will travel through tissue, and that may be altered by things such as salt water and the amount of blood and even autolytic changes. But you can certainly help the pathologist by doing that. One thing you can also do is for the lungs, you can infuse formalin into the airways. Okay? And the eye as well. So the eye is difficult because a dolphin eye is big. What I typically do with any of my necropsies is I will inject a small amount of formalin without putting too much pressure into the vitreous chamber. You'll feel it get turgid. So it'll become harder as you get the formalin in there. But that'll help with preservation. But it also can cause damage or it can cause the retina to lift up if you're not careful. It's personal preference in a lot of these cases, but since I do both the anatomic, the gross pathology and the microscopic pathology, I know what I like. Okay.
Yes.
Any other questions? Yes?
Yeah, I had one related to the previous question. You mentioned tuberculosis in elephants, and I understand in cetaceans and dolphins and whales may be important too, or brucellosis.
Brucellosis, absolutely.
Have you seen any of that?
Yeah, so we certainly studied brucellosis when I was in the residency. I haven't seen brucellosis in any of the animals I've looked at. In all honesty, I've not seen, I've not ever been part of a large stranding, and I've never been part of an abortion storm. Typically what I've done, my work has all been on individual animals here and there. So, but that's a very good point. Brucellosis is certainly something to be concerned about. And there's always going to be zoonotic potential associated with the majority of the brucellosis agents. You know, it's again personal protective here. If you're asking in terms of safety, it's really, you know, it's really an issue of being careful. And you know, the thing is, have you all seen tularemia in an animal? You know what tularemia looks like when you open up an animal? You're going to have this miliary hepatic necrosis. But tularemia is very infectious. Okay? It's a very dangerous disease, and it can be aerosolized. So in the United States, rabbits carry it, and often we'll see people mowing their lawns. They're mowing their lawns with a lawnmower, and they hit a rabbit. And the rabbit explodes, and people suck in the particles of the rabbit, and they can get tularemia that way. But the thing is, when you open up an animal that has tularemia and you see a liver that's full of necrotic dots, you need to be thinking about those diseases. And so if you see an abortion storm in dolphins or seals, you need to be, brucellosis needs to be on your list of differentials, and you may take additional measures of protection. You know, another thing that I do wear that I didn't mention is an apron, which is a good idea to protect yourself from some of that splash, but you have to be careful, and you have to protect the people that are working with you. Your first responsibility is to protect everybody who's working on that necropsy. Does that answer your question? Okay, thank you. Any other questions? All right, good. Only 200 more slides. Oh, sorry.
Regarding number one, dilution of formalin, which is very important.
Yes.
10%, because normally in the field, and the hassle of just getting there, accidents have happened in which they just throw it into pure formalin, and that doesn't work. And then second of all is the proportion of formalin to size of tissue, which is extremely important.
Right.
In
So we say 10 to 1. Formalin to tissue, and you have to basically gauge that. It's never going to be exact. I've never actually made my own formalin, so I've always been fortunate enough to have technical staff that have helped me with that. So I can't answer the issue of the actual how to do the dilution itself, but 10 to 1 formalin to tissue is the ratio, and it's again to ensure that you're not doing, wherever Professor Cifuentes is, the necropsy in a bottle, you know. Okay? Good? Yes? Going once? Going twice? All right. Do you have manatees or duogon here? Okay. One second, please. Okay. So this is a dermatitis. It's necrotizing, focally extensive, maybe marked, and this is associated with a herpes-like virus that was described in 1989. It wasn't seen in a lot of animals, but it's been seen in particular with the reproductive tract in belugas. Now, belugas are a unique group of animals, and we have a lot of data in terms of belugas. If you do a search for tumors of marine mammals, the vast majority are going to be belugas. And so that can lead you to believe, well, then belugas are just far more susceptible to tumors. And that may be true. And because of some of the environmental factors, it actually, to a certain extent, is true. And we'll talk more about some of the environmental contaminants. And even when you have bottlenecks, in populations that could be true. The belugas live in the St. Lawrence estuary up in Canada, and it's a condensed area that's highly polluted. However, does that mean that belugas simply genetically are more predisposed to developing tumors than blue whales? How do we know? We don't see the blue whales. Blue whales may get tumors all the time. So there was a real belief for a long time that sharks and other cartilaginous fish did not develop tumors. And so part of the practice of eating shark fin and actually taking shark cartilage medicinally was the belief that it would protect against tumors. And that's not true. I have seen many tumors in sharks. I've seen and will publish on a few tumors in stingrays. And part of the issue is we simply didn't have a big enough data set to understand the pathology of that species. But because beluga whales are in a concentrated area and actually are also in captivity, we know more about them. We know more about bottlenose dolphins, probably, than, say, the pink humpback dolphins from Thailand, because there are simply far more of them in captivity. We know more about the pathology of dogs and cats than we do about wombats or some other exotic animal. Simply because we see them all the time, we have access to them. And so please remember that. Don't think that simply because there is a statistical variance that appears to highly predispose one species versus another to a disease, it's because empirically that's true. It may not be. It may simply be what we have exposure to and what we have data on. But belugas actually do have some environmental issues that predispose. In the water? In the blubber, I don't know about, but certainly we have some data that suggests increased number of tumors associated with chemicals in the water. Is that me? Okay. And I'll show you some pictures of that later on. Okay, so what we have seen though is the herpes virus that's affecting the reproductive tract. So penile and vulvar lesions in belugas has been reported and is believed to be associated with a herpes virus. And so they're proliferative and necrotizing lesions. Okay, and then we'll talk about the herpes virus, the odorated herpes virus in sea lions later on. Okay, this is a single case report of a gamma herpes virus in the penis of a Lanesville-beaked whale, and it was interpreted to be as a papilloma with chronic ballontitis, and this was identified to be a gamma herpes virus. But it's only a single case. Manatees will develop something called papillomatosis. They're one of the more endangered marine species that we have in our coastal waters in the United States. And the species is typically naturally resistant to infectious disease, but there are a variety of reports that talk about papillomas causing cutaneous lesions in the species. It's potentially of concern if the animals are becoming immune compromised because of pollution, but we don't know that there's a direct correlation. And this is a specific type of papilloma associated with manatees. We talked about sealpox. So this is a parapox virus. It is relatively common. It causes a necroproliferative dermatitis in seals and sea lions. And it is a zoonotic disease. It's typically seen in rehabilitation centers in the people that are working directly with the animals. And the presentation is very similar to ORF that you see in sheep. Okay? And so this disease has been identified in both northern and southern Atlantic and Pacific Oceans, and to include animals off the coast of Antarctica. Okay, so it's very widespread. Microscopically, it's the same thing as we see grossly. It's a proliferative and necrotizing lesion, and there are believed to be 8 species of pinnipeds that are particularly susceptible. Those are gray seals, harbor seals, harp seals, northern elephant seals, northern fur seals, California sea lions, stellar sea lions, and South American sea lions. Okay? Okay. And the infections are often seen in colonies where there are exogenous stressors such as overcrowding, pollution, food shortages, or concurrent infection. These can often be self-limiting and heal, but depending on location, they can also interfere with feeding, as in probably with this animal. And so you can see death associated with juvenile animals that are simply not getting enough nourishment. What is this disease called? Nope, but that's good. I'm glad you know that. But that's not that. It is a pox virus. Tattoo. Thank you. Good. This is called tattoo. That's the colloquial term, but it is a dolphin pox. This is also a dermatitis, although in this case it's predominantly necro-erosive. And there'll be these coalescing spots that start off as just sort of pinpoints, but then enlarge and coalesce. Okay? They're not particularly problematic overall. They're primarily reported in captive dolphins, so in dolphinariums, and predominantly within bottlenose dolphins, although they've been reported in some other species as well. It's a very common lesion, and there's one study that says that there's been upwards of 60% of captive and wild populations of dolphins that have been seen with this lesion. I will say that there is one Chilean report where there's some beautiful photographs of blue whales that are breaching. I don't know who the author is, but it's a very, very nice report, and it is Chilean authors. It's unlimited. It's like an electric and you can see from a distance, the photography shows lesions that appear to be similar to this. We do not know that it is a viral lesion, but I think we should at least have that on our differential list for those species as well. Right, but this report specifically suggests that they look like they're erosive. I can't disagree with you because I don't really know, but that report from Chile suggests that. It's not work that I've done. Okay, and here's another example of it. This is more of a progressive lesion in this animal. There is one report that was put out recently by SeaWorld pathologist Dr. St. Ledger, who is one of the more extraordinary veterinary pathologists who has worked with marine mammals. She's amazing. If you get the chance to hear her talk, I highly recommend it. But Dr. St. Ledger has been involved in a study that looked at the effect of water temperature on the prevalence of these lesions. And what was found is that we'll see a decrease, a reduction in the lesions with increases in water temperature. Okay? And specifically, it looks like you had total resolution of lesions with an 8 to 10 degree increase in water temperature, which is fascinating. There are other viruses that have an effect, that are affected by temperature, but a lot of those are typically in fish. And frogs actually have some viruses that are highly affected by temperature. But this is fascinating work.
Sorry, you are talking temperature in Fahrenheit or in Celsius?
Sorry, probably in Fahrenheit. I apologize. Yeah, I think we're the only country in the world that uses these ridiculous measurements. Okay, and a more progressed lesion here of tattoo with real coalescing. But there's another pinpoint here, probably there, that are also lesions that are starting. These don't typically cause problems in the animals, though. It's more of a, we know it's a pathologic change, but it's not one that is necessarily causing morbidity in the animals. Okay, tissue from a pig. Why did I say that? So do you know what this virus is? This is a calicivirus. This is San Miguel sea lion virus. Okay? And San Miguel sea lion virus is indistinguishable from vesicular exanthema of swine, of pigs. I have no idea how it got from the pigs into the sea lions or vice versa. But it is believed to be the identical virus between the two. But it can cause extremely severe lesions, typically in the oral cavity and on the flippers of the affected sea lions. We've also seen a similar calicivirus causing lesions in dolphins, which is believed to be related. So, vesicular exanthema of swine is one of those 4 vesicular diseases of domestic animals that we worry about. Foot and mouth disease being another one. The others aren't of concern here, but vesicular exanthema of swine presents with very similar lesions. The blistering, which become ulcerative and can cause problems in terms of eating, and then those very severe lesions that we saw here. And if you're going to describe this, you need to tell me that it is not just a dermatitis, it's also a rhabdomyositis, it's affecting the muscle, and it's probably affecting the bone. So it's an osteomyelitis as well. And I'd hate to think about the secondary infections involved in that. Okay, more progressed. You can see it's also here, on the outside. And then we actually have erosion. Too bad this, can we turn off the lights for just 1 second? It feels really dark, but I want you to really see that. Or at least the lights further. Can you see that? You just got sloughing of the skin. Okay, thank you. Okay. Again, this is probably an earlier version, and then these will coalesce and it'll start to slough. This is the tongue, so this is a glossitis. And then in the dolphins, okay, similar presentation. Okay. All right. This is a fibrinopurulent pericarditis, and in this case, the etiology is not known. However, there was one nice study that looked at the most frequent isolates from of sea lions in general. A bacterial isolate, there were E. coli, Streptococcus viridans, Listeria, beta hemolytic strep, and Enterococcus. That's in general. In terms of meningitis and encephalitis, the most common brain isolates were Enterococcus, E. coli, Klebsiella, and Pseudomonas. This is a hypopion. It's, at least in this case, unilateral. I don't know what's on the other side. But the most common ocular isolates were Pseudomonas, Enterococcus, Strep viridans, and E. coli. And then in peritonitis, this is a peritonitis, it's suppurative. There's probably atrophy of fat as well. And in rehabilitation in general, we're going to see those a lot of those gram-negatives. Salmonella, and then Klebsiella, Pseudomonas, Aeromonas, Proteus, Staph aureus, things like that. Okay? All right. This is an embolic meningoencephalitis, and it's hemorrhagic in an elephant seal. And we don't know the etiology specifically of that, but it's likely to be something along the lines of what I just mentioned in the sea lions. Okay. This is a case of salmonella that's affecting the lungs. So this is pneumonia. It's embolic, multifocal to coalescing severe, and this was determined to be associated with salmonella. What disease is this? This is a tough one. Can we turn off lights for a second, please? Thank you. I appreciate it. I'm sorry to make you jump up and down.
Here we go.
There you go. So this is erysipelathrix rhiziopathiae. Thank you. You can turn on the lights. So what do we call in domestic in domestic veterinary medicine, what is the colloquial name for this? It's in pigs? Somebody is? Diamond skin disease. What did you call it in Spanish? Yeah, so the colloquial name is diamond skin disease. Okay? Okay. The organism is Erysipelathrix rhiziopathiae. Okay. What other animals are affected? So we've got dolphins and pigs. What else? I don't think of cattle as getting this particularly. So the main ones I think of are going to be dolphins, humans, pigs, turkeys, and dogs. In the turkey, you see infarction of the wattles and the snood. They have this piece of skin that hangs down from their cere. In dogs, you can see cardiac lesions associated with Erysipelathrix. I think you can see the same in humans. And then in pigs and dolphins, you see these cutaneous infarcts. All right, these are cutaneous infarction, which, because of the way the blood supply feeds the skin, it creates that erosive lesion, which could eventually become ulcerative. And so this is actually a vasculitis, which is affecting the vessel and then causing that cutaneous infarct. And then they can progress as they heal to scarring down quite badly.
Disculpa. Nuestro asistente no puede escuchar.
Do you hear me? Yes. Would you think that these lesions appear in all these different species that are completely different genetically speaking, because all the rest of the species still haven't been studied and they also get the disease? Or would it be some weird evolution that went on there with these species?
That's a very good question. And I'll tell you, when I teach fish pathology, how many species of fish are there in the world that we know of? There's about, well, anywhere between 30 and 40,000 different species of fish, depending on who you talk to. There are probably 30 to 40,000 different herpes viruses of fish, and 30 to 40,000 different types of iridovirus of fish. So it's very possible that there are other species out there we simply don't know about, and certainly when you start looking at exotic species, I think by now we would know if it was a major disease of, say, cats or horses. We certainly describe it in those 5 species. We know it's a human disease. So whether or not it's simply an ancient bacterium, probably somebody has looked at that, done the phylogeny of it, but I don't know the answer specifically in terms of it. I'd be guessing. But I do feel that simply because we don't know what other species are affected does not mean that there can't be other species that are actually out there. Simply because we don't have the data doesn't mean it's not a possibility. In fish, if there's 40,000 viruses, then every single one of you can go out there and publish a paper on a new virus of fish. Easily. And you know, I'll actually show you, I've been involved in description of a novel type of chlamydia and a novel herpes virus. 2 papers that I've published. Or one that's published and one that I hope will be published. Alright, so this is a kidney in an elephant seal. And this is an early lesion at this point. I'll show you a later lesion in a second. So these are called renicles. And I'll tell you, you shouldn't have quite this much white separating them. There isn't that much fibrous connective tissue between them. So what do we see in seals that can be associated with tubular injury? What kind of bacteria? Leptospira. So yes, these animals, so what do you know about elephant seals? Do you have elephant seals down in Chile? Okay, so elephant seals were almost hunted to extinction. They went through a bottleneck, an evolutionary bottleneck, and like cheetahs and other animals, they tend to be more susceptible to disease. Also, as we have colonized coastlines around the world, these animals will often, when they get together, they will concentrate in huge, huge masses. And not too long ago, a few years ago, there was satellite photography of elephant seals and sea lions in giant colonies all together in numbers that had never been seen before. But it was a problem because it's easy for them to transmit. If they're all on top of each other and they're peeing all over the place, then they're transmitting potentially leptospira to each other. This is a very elegant study that was done in sea otters quite a few years ago, but it looked at the differences in pathology between sea otters off of the California coast and the Alaskan coast. And it compared disease prevalence in those two populations. And what it found is because of the pollution and because of the effluent coming from farming into the waters off of California, that the sea otters off of California had far higher rates of leptospira, of toxoplasma, and I think maybe I don't remember the other ones, but at least it was toxoplasma and leptospirosis in that population in comparison to the far less populated areas off of Alaska. And so it suggests that there is very much an environmental impact. And you also have to remember that some of the, like Leptospira pomona or Leptospira griffitifosa, griffitifosa in particular is carried by rats, right? So it's transmitted through the waste of rats. And if that gets into the water through sewage or effluent, then it can get into those coastal waters and infect the animals that are living there on the coast. Okay? It was a very nice study. Toxoplasma gondii. Okay, this is a more advanced lesion, and you can see how we have far more fibrosis that's occurring here with chronicity in this animal. But leptospirosis is a major problem of elephant seals, and certainly is a concern. So in the early stages, we're going to see swelling and some hemorrhage within the kidneys. The urine is going to potentially have that port wine or bile-colored look to it. And then with chronicity, you'll see the kidneys shrink, they'll become more firm, and they'll have this increased fibrosis around them. Okay? All right. I don't know the etiology here, but I did this necropsy, and what I can tell you is the story behind it, which is important. This is sort of a disease that could fit between bacteria and probably parasites. So this animal stranded along with another animal together, two of these pygmy killer whales stranded together. And they were taken into a rehabilitation center, and they had very, very heavy worm loads in their lungs. They had very heavy lungworm burdens. And the wildlife rehabilitators treated them with heavy doses of dewormer. Probably too much. And didn't go through a process of allowing them to clear what they had. And it ended up killing the massive load that these animals had. And those started to necrose. The dead worms started to necrose inside the airways, and then ended up allowing for bacterial infections, secondary bacterial infections. We had a very, very severe suppurative bacterial pneumonia when I looked at this under the microscope, and I believe it to have been associated with the death of the parasites based on the presentation, because both animals had similar findings. And they just went downhill very quickly because of that. Even though best intentions of curing these animals, they had also been pretty beaten up in the stranding process. So, they were immunosuppressed, there were other factors, but the heavy worm burden probably contributed significantly. And then when the ivermectin or whatever the dewormer was that was used killed all the worms, it allowed for this necrosis and bacterial infection. Okay? Okay, polar bear. Not snow. This is a polar bear in a zoo. I don't know where. This is a disease we see frequently in horses. And it's a disease associated with warm temperatures and rain. So this is Dermatophilus congolensis. Okay? It's also called rain scald and a few other things. And it's because this is a species that probably shouldn't be out of a cold and dry environment. unfortunately with zoos, we often see these kinds of unusual atypical lesions. So this is not something you need to worry about in terms of doing wildlife work. This is a zoo problem. Okay. Chile is famous for its penguins. What happens to penguins when we take them into warm environments? Aspergillosis. What else though? There's another fungus that's even more common. Candida. Candida affects penguins very severely. There's one other disease that we worry about. You don't have it here in Chile. Why am I here in Chile? To lecture on malaria. So avian malaria is a big problem in penguins. You do? I thought you didn't have malaria. Okay. All right. Well, so it's a big problem in penguins, and it's going to be a big problem in areas though where the vector is able to survive. So I don't know anything. I apologize. I don't know anything about the environment your penguins live in. If it's very cold, then you probably don't have a lot of vector there. But if you take those penguins from there and move them into zoos in warmer parts, like in Santiago, then where you potentially have the vector, and I don't know the answer to that either, then potentially you could see spread of avian malaria that way. Okay? But Dermatophilus congolensis. So, you know, it's most common in cattle and goats and sheep and horses. We can see it in pigs. Rarely you'll see it in small animals. And but it's most commonly seen in hot, humid areas that have heavy rainfall. So it's usually a secondary invader. We've seen it co-infecting with different pox viruses, but in this species we shouldn't see it ever. But because of the problems associated with zoos, this kind of thing can happen. Okay, this is a single report of Tizer's disease in Eurasian otter, and it specifically had these corneal ulceration and then edema within the cornea as well as other lesions. There was hepatic necrosis and hepatomegaly. There was severe effusion in the body cavities, but this was interpreted to be Tizer's disease or Clostridium filiforme. Okay. So this should be lung and lymph node. And this is Cryptococcus gattii. This was an outbreak that was seen in a range of different marine mammals in California, or I'm sorry, in Vancouver, in Canada. So in the Pacific coast of Canada and Washington State, which is just south of the Canadian border in the United States. And here we're seeing necrosis, granulomatous necrosis within the lymph nodes, the pleural lymph nodes in this harbor porpoise. Is a zoonosis. The animal's presented with this nodular to diffuse granulomatous pneumonia and mediastinal and lymphadenitis. And then in severe cases, we could see retropharyngeal and mesenteric lymph node involvement as well. Historically, this is a disease that's found in association with eucalyptus trees and some other trees as well. So it's very interesting that we had this outbreak that got into the water and allowed for colonization of a variety of species. But this is a zoonosis. Here it is open. Okay? Tissue from a small child who is slightly immunosuppressed. How many parents? Okay. This is a disease we see in the mouth of children. We see it in horses, in young horses. This is thrush, Candida albicans. But it affects actually the blowhole in the case of these animals. Okay. So it's speculated to be more of a disease that we see in rehabilitation centers associated with prolonged antibiotic use. Okay. But it can also be associated with a high degree of stress, so concurrent illness or something along those lines. And you'll see these cutaneous or mucous membranes that form in the oral cavity, the esophagus, and the blowhole. And so these are white pseudomembranes and underneath it Underneath it, you can have erythematous and ulcerated tissue with severity. Okay. Here's another example on the tongue and the oral cavity. Okay. So we have two major diseases that I would think of as being zoonotic in dolphins. One was erysipelathrix, although I don't know about the ability for dolphins to transmit it to humans. And this is the other one. And this disease only affects humans and dolphins, and nothing else. So, who knows? Lobomycosis. Lacasia loboi. Okay, it used to be called loboi, now it's Lacasia loboi. This is a fungal disease that causes a very, very severe hyperplastic change in the skin. The skin of dolphins is already very thick. When you look at this under the microscope, it just creates these very, very deep reefed pegs into the underlying tissue. And you can see the fungus within it. It's thought to follow exposure to contaminated soil or vegetation, and typically associated with a penetrating injury, so some trauma. The animals can become quite debilitated and they can die, typically from secondary bacterial infections, because what will happen is as that skin gets thickened, it'll fissure, it'll crack, and you get these deep cavities that go down, and you get secondary bacterial infections associated with that. Okay? With chronicity, you can see this kind of lesion as well. But and so you see those fissures that can be infected with a secondary invader. All right. Microscopically, there'll also be a lot of granulomatous inflammation that can be associated with in the dermis, and then these, as I said, acanthosis and hyperkeratosis, and these deep pegs of epithelium that are running down into the underlying tissue. Here's another example. Up close. Last one. Okay. This is a dermatitis, necroulcerative, multifocal coalescing, probably severe in this manatee, and this is associated with either zygomyces or aspergillus. Probably associated with immunosuppression as well, because, as I said, these animals are typically quite resistant to disease. Okay. So, sea otter. Has anybody worked with sea otters? No reason we couldn't see this in a domestic animal. This is coccidiomycosis, disseminated in this case. So we've got a severe hepatitis with miliary necrosis and granulomas. Miliary sort of little granulomas under the microscope. Those are going to have yeast in them as well. Might have a splenitis here, maybe a pneumonia, a little all over the place. Okay, I love this picture. Poor polar bears. Poor polar bears. They just don't, they never get a break. So, what color is this polar bear? It's green. What species of polar bear is green? There is no species of polar bear that's green. What do we know about the hairs of polar bears? You know what? Do you know anything about polar bear hair? The hair follicles are hollow. Okay? The hair follicles are hollow, and that helps them keep warm. It creates insulation. But the environment they're supposed to live in is dry and cold. This polar bear, I believe, was in a zoo in Japan. And I don't know what part of Japan, maybe somewhere in the south. And this is called algal trichofolliculosis. So there's algae growing inside the hair follicles of this polar bear. Now, I don't believe that this is actually causing any severe morbidity in this animal, but it's obviously needs to be considered pathologic. Okay. And it's just an amazing lesion because of the anatomy of the polar bear and the environmental conditions within which it is. And obviously in a polar bear enclosure you're going to have water, so they get in the water, they don't dry out, and the algae is growing inside their hair follicles because they're hollow. Okay, now, there's lots of parasitic diseases. So what I'm going to do is, as I go through these, I'll probably list a lot of the names. I don't need you worrying about them. How many of you are going to become parasitologists? Nobody. The other thing is the parasitologists change the names every other year. I don't need you memorizing the majority of them, but you should have a sense of what they are in terms of general categories. And so there's some important ones that I'll talk about. All right. What do you think? So this is probably eosinophilic rhabdomyositis. And inside the myocytes, there are protozoal cysts. What do we call this? Sarcocystis. Okay. Tissue from a sheep. Tissue from a duck. Okay, sarcocystis. Nothing special. Tissue from a dog. All right. Who's been bitten by a mosquito? Dirofilaria immitis. Okay. Although in this case there also could be Dipetalonema spirocauda, but Dirofilaria immitis is found in seals. Okay. This is Acanthochilionema odentali, and it's non-pathogenic. It's just found living in the pericapsular tissue of the kidneys. Doesn't cause pathology, it's host adapted. Does anybody know ganglenema? You know ganglenema in the esophagus of cattle? Have you seen it? If you see it, when you're doing a necropsy of a cow, you look in the esophagus, and you get this beautiful subepithelial little line, perfect like that. It's not causing any inflammation, it's not causing a problem, but you need to know that it is actually a worm that's causing that effect. Sorry? That's a good question. I don't know the answer, I'm sorry. I'll find out for you. But make sure that, Professor, I need to answer that question.
I don't know. What question was that?
If these are adult or larval worms.
No,
Okay?
Just wanted to know.
Okay. I love sushi. I eat a lot of sushi. I probably shouldn't. Luckily, we flash freeze our sushi. But this is anisaki's, okay, which has the capacity to transmit to humans. All right. You typically don't see direct seal to human transmission or other marine mammal zoonotic transmission. It's through fish hosts. I have seen it on necropsy of fish. You have to be careful. But this is a very severe lesion. And what I want you to pay attention to is, you see these ulcerations and erosions? Okay? The whole thing is thickened. It's angry. There's a catarrhal gastritis here. All right? You could probably say it's eosinophilic. You could say, certainly, that you've got severe erosion and ulceration from the physical presence of these worms. Okay, but this is a quite severe case. This is from a museum sample, and this is gastric nematoidiasis, and there's a few different ones that it could be. It could be contracecum, it could be pseudoterranova, or Foascaris are possibilities. I don't know which one this is, but it's a nice lesion. Now this has been fixed in formalin, or in alcohol, I don't know specifically, but it's a museum specimen. This is gastritis again, necroulcerative. There's a hyperplasia in here as well to the epithelium as it's trying to protect itself and react. This is quite severe. And there are lots of nematodes in this lesion. And again, I'm not sure which ones these are. Actually, no, wait a minute, I'm sorry. My slides are out of sync. This is acanthocephalids. These are acanthocephalids. So these are thorny headworms, and their heads are buried in the mucosa. Okay? So they're actually causing the damage because of the physical process of burying their heads into the mucosa and feeding off of it. Okay, this is another case of anisakis. So this is a sad story for a pathologist. But I did a necropsy on a dolphin, and as I was peeling the blubber away, I saw this kind of a lesion. And it's a great lesion. And I didn't know what it was. And I actually got the worm, which was a long worm, and I was able to pull it out, and you still saw this fibrous tract that it was living in. But I didn't take a photograph. This is not my photo. This isn't the animal that I did the work on. This is crassicauda, and it'll be in the subcutaneous tissues. It doesn't cause a lot of reaction, but if you remove the actual worm, you will see this fibrous tract, almost like a straw, that it's created as it's moving slowly through the tissue. It's also seen significantly in the mammary glands, or mammary tissue of dolphins. Okay, and there it can cause a little bit more reaction. But I wish that I had had a good camera and taken photos. Okay. So all right, so your guys' symbol, for those of you that are in the vet school. All right, the staff, what is it, the staff of Asclepius, is that what's called? So there's different theories in terms of where this came from. One theory is that, and the theory that I like, whether or not it's true, is because I grew up in Africa. So I spent most of my childhood living in the Sahara Desert, and there was a worm that would embed itself into your skin and go up, and it would cause extreme pruritus, horrible pruritus, horrible itching. And they called it the fiery serpent of the Nile. The fiery serpent of the Nile. And it's dracunculus. Okay? Exactly. And the way you get it out in Africa, because the female will stick its tail out and ejaculate the eggs into water. And people will be, they're itching so badly they put their feet in water. And the intermediate stages require invertebrates. So what they do is they get the worm to stick itself out, and then they clamp down on the end, and slowly, very, very slowly, over many days, they slowly turn that piece of wood and wrap the worm out until they get it out of the leg, because if not, it'll break and it'll die, and then you get necrosis inside. Okay? I don't know if it's true. I like the story. This is Dracunculus lutrae. Okay? We see Dracunculus also in dogs. This is Dracunculus lutrae, and it can cause similar type of lesions. This is subcuticular, but it causes severe pruritus, and areas over the skin can be denuded. They can lose their fur and become hemorrhagic as well. And that's in part because of the itching. Eventually you could have erosive lesions and ulcerative lesions too. Okay, so the next two parasites are quite important as you do necropsies. So you can't see the worm here, but it's in there. And this is Paraphiloroides decorus. We see, what we're looking at here is a pneumonia. It's eosinophilic and edematous, and it's probably occurring throughout the tissue. This is a parasitic nematode that's found within the respiratory tract of pinnipeds. We see it in California sea lions and stellar sea lions. You'll see it in northern fur seals, South American fur seals, Antarctic fur seals, and South American sea lions. It's seen in the whole Pacific, And in particular, we see it along that Pacific coastline of the United States. And it's considered to be a common cause of strandings of sea lions in the U.S. at least. And you can get extremely heavy worm burdens in these animals. The prevalence is highest in young sea lions that are less than 2 years of age, and it can be due to the immature immune systems, but it also could be due to the fact that they have that they're feeding off of tide pool prey, which is believed to be the intermediate host. Okay. It is also felt that there's a possibility. Oh, I lost him. We have to tell him when he gets back. Okay. It's believed to be potentially a vector for brucella. Okay. Then another parasite that we see is Otostrongylus circumlitis. And in that case, typically it's actually in the arteries, so you get an arteritis, but they can pour out into the heart as well. So sorry, this is Otolitis circumlitis, and this should be Paraphiloroides decorus. But this is going to be an arterial nematoidiasis. They've opened up the heart here, and you've got all these worms living inside the large arteries. Okay, they're commonly reported in the lungs of harbor seals, and then they're also called the heartworm of seals and northern elephant seals in particular. This can be a pretty common finding, particularly again in juvenile animals, and you can see as they spill out and get into the airways as well, you can see epithelial hyperplasia from the presence of the worms. Otostrongylus circumlitis. So, these are acanthocephalids again. So it's a similar thing where they're burying their heads into the mucosa. In this case, the intermediate hosts are crustacea or various larval insects. And there's a variety of different species. You can see ulceration and even perforation of the gastrointestinal tract associated with these because of the way they feed and the way they latch on. And you can also see obstruction of the bowel based on large numbers of parasites. It is really warm in here. Is it just me?
Yes.
Really warm. Can we turn up the air conditioning at all? Is that possible? And these are acanthocephalids within the omentum. Okay. If this was a cow, what would this be? These would be flukes. These would be trematodes. So this is a biliary trematodiasis. As they open up the bile duct, we see the trematodes here. Fasciola? I do have the name here. Hold on. This is specifically Zalophotrema. Zalophotrema. So it's not the same exact, but same basic principle. Now, this is my photograph from a finless porpoise from Thailand. And this is called Brownina cordiformis. So this is a trematode as well. These are flukes. Brownina cordiformis. And cordiformis means, either in Latin or Greek, means heart-shaped. Okay. And I think you can sort of see the heart-shaped nature of these. When you look at it under the microscope and cut through it, it's full of eggs. She doesn't like bradycorna. But it's full of eggs. And you can see those trematode eggs, which have very specific look to them with the under the microscope. But this is an interesting lesion, and it hasn't been well reported. Sorry? I'm sorry, this is the stomach. And it's typically seen right near the duodenum. Okay? But finless porpoises are a species in Southeast Asia and sort of the Taiwan waters, Thailand waters, and then towards India. So we see Hong Kong, Taiwan, Thai waters, and then on the other side, between India and Burma and Thailand. All right. So remember I told you when we were looking at the picture of the dolphin, the drawing, that you had to red loaf the blubber towards the tail? You should do it the entire length of the animal, but in the tail for some reason, you see a lot of these. And these are subcuticular cestodes that lodge within the blubber and within the subcuticular tissues. Very common. They often don't cause much of a reaction. Okay? But they're very common and you need to look for them. And you can do that just by bread loafing through the blubber on the tail of the animal. There's several different species that cause that. And here in a fur seal you have something similar. Okay? And this is probably Diphyllobothrium. And here they're causing granulomas as they've encysted in the wall of the intestine. A different species of cestode, but Diphyllobothrium also. In this case, we're looking at Diphyllobothrium 2, and here within the cecal tissue, the cestodes. Okay? This actually is in the snow. But what is the species? What are we looking at? What disease? Tissue from a wombat. So it's the Sarcoptes scabei again. Okay? And it's going to look exactly the same under the microscope as we see in a dog. Okay? So hyperkeratotic lesions with the mites that are buried in there. Okay, this is actually the nasal mucosa. This is inside the nares of the sea lion. And these are mites. These are nasal mites. This is causing a mucoid rhinitis, and these arthropods are in there. And this one in particular is Orthohalorhynchi diminuata. I'm sorry, Orthohalorhynchi attenuata is in the nasal tissue, and then in the upper airways is Orthohalorhynchi diminuata, which is in the airways. But we see a variety of different mites within the respiratory tract of these animals. Okay? Attached here. And they don't appear to be creating too much of a problem, although you can see some hemorrhage and erythema around the attachment point of that animal, that arthropod. This is the nasal planum. And same thing, we've got these nasal mites that are attached, and here it's a little more aggravated. There appears to be some hemorrhage. I'd say it was mucohemorrhagic rhinitis, and it might be, you could even say eosinophilic because of the presence of the parasites. And this is just a different species of mite. These are cutaneous stalked barnacles, and this is something that you will see here in Chile. There's a nice, the next two photographs are from Argentina, and these are just an incredible load of these stalked barnacles on this animal. And certainly the amount is such that it's actually going to impede, I'm sorry, I keep getting in front of you. It's going to impede the ability of the animal to swim and feed properly. Okay. And to a certain extent, this is like the epibiota that we talked about in the turtles and in whales. These other sea creatures that attach on. But typically in this case, you would expect the animal to be immunocompromised or something else going on to allow for this kind of a heavy load. I don't know. Do you know about this type of dolphin? It's not my picture. Is this freshwater? That's interesting because these are barnacles. Estuary? I don't know the geography, but so now I have two answers I need to get you. biota, this is a humpback whale, you'll often see barnacles on them. And this is probably not causing any kind of a problem. And in grail whales, you can see these cutaneous mites. So you need to be looking for these things as you do your necropsy. You don't dive right into the abdomen. You want to take a look and see what is living on the animal. Cyanids? I don't know. I just called them cutaneous mites. Okay. So, this was a study that I was involved in Thailand. And my belief, this is my personal opinion, there's a paper that I've written that's being reviewed right now, so it's not a publication. We discovered in a series of different animals that were both in captivity and in wild populations. In fact, I'll go back, but this is a wild animal who had very similar lesions, earlier obviously than this. These are squamous cell carcinomas that are metastatic throughout the animal, extremely severe, and we did a lot of PCR to try to determine if there was a viral etiology associated with it because it was seen just in this species. So I looked at one dolphinarium where we had several of these animals that were affected, and then we had Irrawaddy dolphins, which are a different species, that were completely normal. The water quality was checked. Potentially, because these are pale skinned, this could be UV associated, but then why is it not something that's already been recognized in the species for a long time? Unless we have something that's changed in the animals, or something that's changed in the environment. And the fact that we see it both in wild populations and in captive populations is interesting. We haven't proven anything. We did find a new alpha virus, and we've described that new alpha virus in this species, but alpha viruses are not typically associated with tumor production. So we're not 100% sure. What we've done is describe the disease, laid out the information we have, and then subsequent information needs to be gathered to eventually prove what is the cause here. Certainly some genetic work would be interesting to do, but this is an endangered species. I will tell you, a lot of you are veterinarians, are going to be veterinarians, and you look at this horrible lesion, and you ask yourself why this animal lived this long. Thailand is 98% Buddhist, and they do not believe in euthanasia. Okay? So they treat they treat with supportive care and painkillers and the like, but they're not going to euthanize. So it's just the way it is. It's a cultural difference. Okay? In this case, the lymph nodes were necrotic, but there was also tumor that had metastasized internally. Okay. And it's important to note from the standpoint of the epidemiology that we were seeing this in multiple different populations of wild animals, and the earlier stages in the captive animals looked exactly the same. So we interpret these to be similar. We hypothesize that these are similar lesions. Okay, liver. If this was a bear, an Andean sun bear, what would this be? So bears are very prone to biliary tumors and hepatic tumors. This is a biliary carcinoma in the liver of this sea lion. Okay, I mentioned that we were going to talk about belugas again. So belugas are in water that is quite polluted, and there has been a correlation made between these animals and anthropogenic chemicals in the St. Lawrence estuary. And this is a serous adenocarcinoma, just like you would see in a cat, right? So the carcinoma is there, the adenocarcinoma is there, and it has so much desmoplasia, or fibrous connective tissue associated with it, that it squeezes down. You expect to see a mass effect when you look at a tumor, but sometimes we see less, not always more. And if you look at this, you're going to see mainly neoplastic tissue. So this animal would have, and you can already see there's a bit of mega intestine here because of the inability of food to pass through. So this animal may have actually perforated and died because of the perforation, and then they found the tumor impeding the motility of the gut. But we do see a higher number of, I think over 50% of all tumors in marine mammals have been described in belugas. And so, again, that raises the question, is it because they're genetically predisposed? Is it because they're living in water which is highly polluted and therefore they're being inundated with products that are stimulating neoplastic progression of lesions? Or is it simply because they are close? We have a lot of visibility over them. They're very charismatic animals. They're friendly animals. They're used in captive dolphinariums and the like. And so maybe we just have greater visibility. Because I don't think it's responsible to say that they necessarily have a genetic predisposition to it at this point in time without proving that it's not something that exists elsewhere simply based on those numbers. We simply don't know. We don't have enough data at this point. But there are other factors that play an important role in neoplastic transformation in these animals. Okay. Papillomas, nothing particularly special. This, on the other hand, was an interesting study that was done by one of my mentors, Dr. Lipscomb, and his colleagues were able to describe this gamma herpes virus that in sea lions, which was causing metastatic carcinomas that were primarily in the genital tract of these animals. The study that he first published looked at 10 animals, and you had 5 out of 7 of the females had tumors in the cervix. 5 out of 7 had tumors in the vagina. 7 out of 7 had tumors in the cervix. 3 out of 7 had tumors in the uterus. And then 3 male animals had it in the penis and the prepuce. Okay. This is associated with the gamma herpes virus, and microscopically you can occasionally see the intranuclear inclusions associated with the virus. And this is called otorine herpes virus type 1. I believe this is, I forget what this is. I think this is a large carcinoma. We're going to skip over that one because I don't remember. This is called carcinomatosis, and this is when you have a carcinoma that ends up spreading and seeding throughout a body cavity. So you can see a carcinoma that may start somewhere, say, in a mammary tissue, but then ends up getting into the thoracic cavity, and you'll see seeding throughout the cavity. And so this is a case of carcinomatosis. There's nothing specific to marine mammals about this. This is the same kind of thing you might see in another animal, and I wrote a paper on a pygmy goat with a similar form of disease in that particular animal. Okay, what do you think? What do you think? What kind of tumor? Meningioma. Nothing special about this. But if we didn't open the brain, we wouldn't have found it. Okay. Congenital lesions, left palate. Normal kidney, hypoplastic kidney. Same thing. Normal, hypoplastic. Okay. This is an important disease. All right. This is domoic acid intoxication. All right, and this is, there's been a series of dangerous algal blooms that occurred in California, which ended up affecting California sea lions. And what you end up seeing is the prey for this species was the northern anchovy, and those fish were accumulating the toxin associated with the algal bloom. Typically what we were seeing was persistent seizures, and then the gross findings were that these animals were dying acutely with piriform lobe malacia in the brain, myocardial pallor, bronchopneumonia, and then dystocia in pregnancies. They were often dying months after intoxication, and microscopically we would see bilateral hippocampal atrophy. And the hippocampus is difficult to see, but it's right there. And it wasn't the entire hippocampus that was affected. It was just one group of cells in the hippocampus, but that was a microscopic diagnosis. But you do have necrosis here. Okay? You've got loss of hippocampal tissue here and here as well. It's bilateral in this case. Here we've got the myocardial pallor. And then sea otters were also affected by this. And you could see enlargement of the heart. Okay. But this was, in sea otters, it's a little bit more complex because the belief was it was domoic acid, but then there was also Sarcocystis neurona, which potentially played a role in sea otters. But we see this cardiomegaly in a way, dilated cardiomyopathy, excuse me. And it was seen in the southern aspect of the range of the sea otters during a specific time of year. So the myocarditis they tried to identify the epidemiology of this. There are a variety of factors. Age seems to play a role. The body condition played a role, and then also exposure to domoic acid and Sarcocystis neurona. So what do you think this is? What? The gastric ulcers. But what else is in this image that might play a role? So some of this is hemorrhage. You're absolutely right. And the good gastric ulcers. Some of this is also oil. Okay. So oil ingestion, you end up with a lot of hemorrhage, and then also this dark brown pigment, which is partially associated with the acid digestion of the blood in those areas of necrosis. In addition to the gastric ulcers, you're going to see emphysema, hepatic lipidosis and renal lipidosis, and central lobular necrosis of the liver as well. Here's the intralobular emphysema, and that's typically because they're breathing in the aerosolized, volatilized particles associated with the oil. And this is when you have an oil spill, such as we've seen far too many times throughout the different oceans around the world. Gastric ulceration here. In this case, this is not associated with oil. This is simply a common finding we see with rehabilitation, and it can be associated with antibiotic use. It can also be associated with steroid use, and so antibiotic use disrupting the normal gastric flora, but then steroid use, and then just stress or concurrent disease. It's really going to be multifactorial, and there's nothing specific associated with it. I think this is a fascinating case, but we don't know specifically what it is. We would see similar things in animals that have liver disease potentially, and so photosensitization, this could be associated with that. It could also be a severe sunburn, or it could be associated with infectious disease too. Or autoimmune disease, sorry. Okay. What disease is this? What's the tissue? Muscle. What color is the muscle? White. Just play with me. It's white. So it's white muscle disease. What is white muscle disease caused by? Give me two different possible causes. I think I heard vitamin E, selenium, somebody said. Okay, vitamin E, selenium imbalance is one. What's the other one? Capture myopathy would be a good one. There's other causes too. But capture myopathy, it's a wild animal, the stress associated with it, trying to escape. This guy was swimming away from a killer whale too fast and ended up causing damage to himself. But yes, capture myopathy, vitamin E, selenium imbalance would be certainly possibilities here, white muscle disease. All right. This is probably a traumatic injury, but this is a progressive arthritic change with fusion of the bone, and here an osteoporosis with also just compensatory fusing of the bone in the vertebrae. Cataract. Okay. Here we've got hemorrhage in the eye. What would cause this? Trauma. It's a boat propeller. unfortunately, this happens far too commonly. I don't know if you've been following. There's been some recent reports about the size of the trash island in, I think it's in the deep Pacific right now. It's bigger than the size of the state of Texas, which is one of our biggest states. It's huge. It's huge. Okay, what caused this? Tell me. Excellent. Well done. This guy did. Well done. She gets special credit. Yeah, that's fantastic. So these are predation lesions for the most part. This animal may have been alive when they started to feed on it, but if so, it would have been extremely debilitated. But often this will happen post-mortem as well. But these are cookie cutter sharks, and they'll lodge onto it and then they'll chew it off. But that's a relatively common lesion you can see in a stranded animal. Okay? Depending on whether or not you have these guys in your particular area. And there's really nothing I wanted to say about this. Unfortunately, there's been far too many strandings of a range of species. And there's so many different possible causes of them. There's been a lot of reports associated with noise underwater, with human activity, even with naval activity in terms of testing of things like sonar underwater. But we don't really know. Unfortunately, we just see far too many of them. There is one disease that I didn't touch on that was viral that's very important, and that's the morbillivirus. We just don't have any good lesions associated with it for a gross pathology course. It's more of a microscopic lesion. But what are the two diseases that have been eradicated on the planet? Smallpox? And? Yeah, smallpox. What's the other one? Somebody said it. Rinderpest? Who said rinderpest? You deserve special credit. All in the back there. Rinderpest. Do you know rinderpest? You call it something else here in Spanish. It's bovine.
Peste de los pequeños humeantes. De los pequeños humeantes.
No, it's not PPR, but it's related to PPR. It's not peste petit ruminant. But you're absolutely right. It's the same type of virus. Peste petit ruminant is a morbillivirus of goats and sheep, but rinderpest was in cows. But you call it something different here, like bovine fever or something like that. I don't know. But it's an important disease to know because it's one of two diseases to have been eradicated from the planet, and it's a veterinary disease. Number two, the first school for veterinary medicine was founded in France during the Napoleonic era with two purposes. One was to create people who could effectively take care of the cavalry horses, and the other purpose was specifically to try to eradicate rinderpest, because every 20 years the virus would come back because it would be naive animals, and it would wipe out about 80% of the cattle. And they would die from horrible diarrheal disease, essentially. And so it was destroying the European economy every 20 years or so. And so as veterinarians, those of us that are veterinarians or veterinary school, our entire field of education started with the intent of eradicating this. And I think the last time we saw a case was about 10 years ago in Somalia, but it's believed to be fully eradicated. Okay? The reason I mention it is we get a lot of strandings associated with dolphin morbillivirus, okay, which is morbillivirus will affect, you'll see viral inclusions in both the epithelium and in the epithelial nucleus and the cytoplasm. Okay, and it can cause very necrotizing lesions throughout the gastrointestinal tract, and the animals will strand in large numbers, very virulent. Cetacean morbillivirus. Of interest, it's a disease that typically jumps a lot, and so you'll see canine distemper, for example, which is also the same group of viruses, canine distemper. That jumped into the Lake Baikal seals, the same thing, but we also have seal or phosid morbillivirus, which is slightly different, and it causes major die-offs and strandings in these animals. The Serengeti has lost 1/3 of its lions to canine distemper. because it's jumping from the dogs to the lions. So these are diseases that like to move. What is the human morbillivirus? Measles. I don't know what you call it in Spanish. What is it? Paperas? All right. Guys, that's it. So, any questions? You're exhausted. I've been talking for far too long. Okay, before you leave, how many of you decided you want to be pathologists now? Why am I not seeing every single hand? Okay, well I hope a few of you at least are interested in the subject, and please consider it. Federico will have my email address. You're all welcome to reach out to me if you have any further questions. I have some questions that need to be answered. And otherwise, I will send this material to Professor Cifuentes, and he can distribute it to all of you. And I believe you will be able to access the video as well. So if you had difficulty understanding my English, I apologize. Probably better than my trying to do this in Italian. But what you can do is you can compare at a later date at your leisure. Okay?
Bueno, creo que hablo por la mayoría, yo creo que el inglés sí se entendía. Y obviamente agradecemos la visita del doctor, haberse propuesto voluntariamente para vernos. En el camino para acá, antes de tener la experiencia de hablar con ustedes y darse cuenta que efectivamente tuvimos una asistencia bastante buena, ya asomó la posibilidad de que pudiéramos tener algo un poco más especializado, un poco más largo, qué sé yo, etc. Así que si es de interés de ustedes, podemos trabajar en hacer alguna reunión un poco más prolongada, ¿cierto?, en el futuro. Como les decía el doctor, efectivamente los que se inscribieron en la primera ronda de inscripción, y además los que se inscribieron hoy día al entrar, si registraron sus correos, van a entrar en el correo masivo y yo les voy a entregar todos los documentos, los links, y voy a aprovechar de hacerles una pequeña encuesta para preguntarles qué es lo que pudieron haber encontrado bien o mal para poder mejorar esto. Finalmente, afuera en una mesa para esas personas que se inscribieron en la primera ronda están los diplomas para que los puedan retirar, y les deseo un buen viaje a casa.
I'd like to ask for one favor, please. Could everybody come up on stage? We take one photograph as a group.
Una, dos,